barrier and causing inflammation and neuroautoimmune
Sir,
diseases and
already
all.
environment health is included as environment is a part of actually one health. In fact, in Hyderabad, we are working with the Telangana UPTRI, that is the Telangana State Environmental the Training Institute. Particularly, they are working on the project of Mushi River cleaning. We are part of it. We are part of it because lot of slaughter waste, other things are going there. So definitely, environmental contaminants, pollutions, even heavy metals. Actually, we found out lot of contaminations. So how it comes, how it can be minimized? We have a role there, definitely. Secondly, many of the studies that are being done in animal population. Any product, something comes actually. Always do toxic study in animals first. So unless veterinarians are there, how they can do it? That's why this institute I mentioned in Hyderabad, ICMR, they are working on this aspect also. Thanks.
Thank you.
Yeah. Okay, thank you very much. Yes.
Thank you very much, sir, for your insightful and inspirational session. I would like to call upon stage Dr. Rajpal Singh Kashyap, sir, to felicitate Dr. S.B. Barbudde, sir.
Now, I welcome Dr. Pragya Yadav, ma'am, India's frontliner virologist and pillar in high-risk pathogen research. Dr. Pragya Yadav from ICMR NIOH Nagpur is one of the country's leading experts in biosafety, outbreak response, and handling high consequences pathogen. She has been instrumental during several national health emergencies, providing scientific leadership and rapid diagnostic support. Her expertise in BSL-3 and BSL-4 virology has greatly strengthened Indian disease preparedness and response strategy.
Presentation is missing somewhere. In the meantime, they'll find. कैसे करोगे? नहीं, उनको फोन मत करो, लगा दो ना. So any, I can talk without presentation. You go down and bring him if possible, otherwise fine. Anyway, good afternoon, everyone. Thank you for joining post lunch, especially the students. Hello, everybody who are all sitting behind. Why you don't come in the front? Why you are hiding in back? All the students, please come forward. This program is for you. So I request all of you to come forward. So we just had Dr. Naveen and Dr. Barbudde a very good presentation on pandemic preparedness and also how they are working on One Health. So I'm going to talk to you, and if you come forward, then we can do an interactive session as well. But if you are hiding, then I have to keep speaking on this subject. So how was how was your competition? Did you enjoy? I want to hear more loud. Did you enjoy it? Okay, so Josh is very high. I'm very happy to hear that. So today, why this activity was initiated, if you give a thought about it, it is because we wanted to create some awareness among the young youth. Because when we launch this National One Health Mission, we expect the young people to know about it. And most importantly, if you don't know what is the vision of our government, how you are going to participate. So, as we are talking about pandemic preparedness, surveillance, disease, diagnosis, I think you heard all these very important words. So, these are the some key words which emerge after post pandemic, and most of the people know that what actually it means. So, I give you a story because I don't have a presentation at the moment, it's missing somewhere between communication. So, if you know that before COVID pandemic, there were many pandemic in the past, and the pandemic started as early as 116.7 BC, which we call as And it was started with the bubonic plague and different kind of plague, then monkeypox, then we have a Spanish flu and so many kind of flu. And every time when these pandemic happen, it cost millions of the life. It affected them, it killed. If you just talk about smallpox, it kills almost 80 million people. So you can imagine a number of people must have suffered and died. And the smallpox, a disease, you all know about the smallpox. Did you take vaccine? So the people who are born after 78, they would not have taken vaccine, because I'm born in 78, so I don't have any vaccination. So I imagine none of you have taken vaccine except the front row. So because the disease is eradicated and there is no vaccine available, but it to eradicate the disease, you know how many years it took? 100 years. Even once we discovered the vaccine, which is discovered by Jenner, which called the father of vaccinology. And in spite of that, it took 100 years to eradicate a disease. So, and the COVID was happened how many years back? How many years back? Just 5 years. So we are running fifth year when the COVID pandemic happened, and we are living a normal life. We don't have disease now, which is affecting you, and we are. But the first 2 years were very, very tough, right? Everybody agree to that? How many of you sat at home during the lockdown? I want to see the hands in the young. Okay, so most of you, right? So you know the suffering, how much it's suffering everybody has gone through when the COVID pandemic happened. What the good thing happened post pandemic? Can somebody tell me? Can you raise your hand? What was good thing about pandemic? Was anything good about it? There is a one thing which you can definitely say, that is a digital innovation, right? Everybody now work on mobile, everything is available on digital media. You did lot of study during the COVID pandemic on mobile, TV screen, and also in the internet media. There is a lot of not benefit as well. But there was some good point which COVID brought, and there are three things which COVID brought which I would like to mention, and you must be knowing. First, that we are a better country who can do diagnosis better, because during the COVID we did lot of innovation which has brought a lot of indigenous technology. And the NIV, Dr. Naveen told that NIV was the one laboratory across the country who are doing COVID testing. Now we have 3,000 laboratory across the country. We have multiple institutions. So that was the first thing, that we enable our country for diagnosis. Second, we enable our industry to produce large number of diagnostic kit. Third, we did so many innovation that we can create any kit today. The fourth and most important, the development which happened having the vaccine platform. Now India can make any kind of vaccine. And second thing, we become vaccine hub because of that discovery, innovation, and our input and our interest in it. So now we have academia ready for innovation. We have industry ready for doing any research and also making any vaccine. So these are the some of very, very important plus point you can say about the COVID pandemic. But we also realized during the COVID pandemic, when COVID started, there was only few laboratories were functioning. And if you want to work on, not COVID was still a BSL-2 pathogen, but there are many pathogen very deadly, as Dr. Naveen must have mentioned about Nipah, Ebola, and so on. If you even just get exposure to disease, the 100% mortality is there. It means and there is no treatment. In the past there was no treatment, there was no vaccine, and that's why they required highest attention, and they called as priority diseases. So there are many priority diseases, and in India we have such very high viral diseases. It's called Crimean Congo hemorrhagic fever. We call Nipah, and we also have a threat of all the time emergence of any disease. But is only viral disease important? No. Bacteria, fungal, and other diseases also very important. In the morning, some senior person said, that the next pandemic would be of antimicrobial resistant, which you all must have had, because I know that in veterinary sector also a lot of misuse of these antibiotics happen. So that is another challenge which India is facing, and it requires a lot of attention. There was another issue which we realized, that everybody work, the veterinary people work and focus on animal health, all the doctors and the scientists, they are working on exclusive on human health. Somebody is expert of earth sciences, environment science, they are working on that field, and we are not talking to each other. So what happened, most of the time we are duplicating our efforts, we are doing the same, we are turning the same wheel again and again, and we don't know the solutions. So the government of India thought during the COVID pandemic, we need to do something better. So they came up with the idea of developing this national one health mission. Under this program is happening, and we are all here. So finally my slides has arrived. So I quickly take you to some of the slide, because I think I have also. So this is a BSL-4 facility which is in Pune, and I'm sure Dr. Naveen must have told about it. So let me take you to quickly forward. So these are some of the diseases which has arrived in 21st century, and Dr. Barbuddhe was also mentioning about it. And there is one disease which I would like to mention during the COVID pandemic. When everybody was suffering with the COVID pandemic, there were many diseases which emerged and spread across the globe. And monkeypox is one of the example, which again proved that disease has no boundary. They cannot be stopped once they start spreading, and it is spread across entire globe, and now it is a major problem among the people who transgender people, MSM, and also the high risk populations. But there is another disease which is spread, Zika, and also Ebola has spread. So this is a picture which you have seen sometime for Ebola, where people are having some PP on, but this is a picture of Kerala, when Nipah outbreak happened in 2018, a large number of people died. And I'm talking about before pandemic, when we didn't have such kind of preparedness, we didn't have mindset, and we were not working together. But Nipah was the first example in where actually all different sectors came together to work to understand about this disease. And that actually helped us to understand the ecology of this disease, the cycle of the disease. You must be knowing that the first time it was discovered in Malaysia, where they found that there was a cycle between pigs, bats, and human. In the Bangladesh, they found there's a cycle between palm sap consumption and bats. But in India, we have missing link, and there is a lot of hypothesis that how this disease transmit. And in India, we don't know. There is still a lot of speculation, but we have done a large study, and we found that virus is present in bats. It's very, very true. It is not only present in Kerala, but present in many states. And we have proven multiple times. But how this spillover happened from bats to human, that link is missing. And there is multiple hypotheses. Some people say maybe some other host is involved. Maybe aerosol is responsible. We still do. And this is a challenge for you to discover that how it happened. So please think about it. But when we came together, state, central, veterinary, human health, NIV, ICMR, everybody, NCDC, DSD, everybody came together, and we could quickly able to detect this disease. We are now having data integration. We have community awareness. We have a very good trained staff across, and so on. And a lot of studies, we are now able to develop the medical countermeasure for Nipah as well. So this is the success of working together, which I put is a very good example for Nipah. When we call about any outbreak, whenever any disease happen, we want to diagnose, especially if it is a disease which transmit very quickly from one human to human, or animal to a human, or human to animal. So you want to contain the disease by detection and by isolation. But that's it not. There could be diseases which could be as a bio warfare. People can target your country by different means, that where the virus, pathogen, bacteria can come in the form of any way, and it can affect you. And you keep guessing it is a natural outbreak, it is a man-made outbreak, as you all know about the COVID, right? there is still a lot of story that how COVID has spread and nobody has clear-cut answer. When we talk about discovery of SARS coronavirus in India, because such a big population, India did a work on it because Dr. Naveen must have talked, so I'm not going to give you detail, but I want to tell you one thing about the risk group of pathogen. What are the risk groups? So there are 4 risk group of pathogen, the group 1, group 2, group 3 and group 4. So group 3 and 4 are those, they are very highly infectious and we don't have treatment and we don't know how to treat the people. So they, and the group 4 is the one where there is no vaccine, no drugs are available and they can be handled in BSL-4 facility. The group 3 pathogens are those for them any treatment or vaccine is available, for example monkeypox, and it can be handled in a BSL-3 or BSL-4 facility. So to have these kind of infrastructure, Government of India thought in the past, and in NIV first we develop a BSL-3 and then we develop a BSL-4 facility, and that actually help us to know about different diseases, and that's how we prepare our country to respond in different public health emergencies. So this is a BSL-1, which is mostly MSc laboratory, if you remember. This is a BSL-2 laboratory, which you have seen in your microbiology, biotechnology laboratory, where you will have sink, you will have dose, you will have biosafety cabinet. So this is a simple structure of BSL-2. When I say BSL-3, it means it should be a unidirectional flow negative pressure lab where you use very special kind of PP and you will have a gradient pressures available in the facility. This is a BSL-4 facility where you will have a laboratory which is highly secure. You will have completely sealed laboratory with the negative pressure, very specialized laboratory, and you use a very specialized PPE, which we call as a positive pressure chute. And they are the most secure laboratory where you can do any manipulation on a biological pathogen and that will not affect you, community, or to your products. And how we do it? We do this personal equipments, we do administrative controls, we do engineering controls, and most importantly, we also follow different practices, and all together we make sure that no pathogen actually escape from such kind of facilities. Post pandemic, Government of India invested a lot. We have now a larger network of VRDS. We have many NGS, next generation sequencing facility, and there is 4 zonal NIV coming, and additionally in Nagpur we will have National Institute for One Health, which will have BSL-2, BSL-3 and BSL-4 facility, and that will help to bring all the stakeholder of National One Health Mission to work together. During the COVID pandemic, as I explained, that we have done a lot of good work and we developed even indigenous vaccines that include a Covaxin, Zydus, Zyco DNA vaccine, and so on. So during after COVID, we realized that we need to work together. And as I was mentioning, that for the future pandemic, how we prepare better. So there are certain thoughts that we should have early detection facility, we should have genomic sequence surveillance, wastewater surveillance, and we should able to assume that what new pathogens can arrive, and we should have infrastructure made accordingly. So National One Health Mission was declared by Prime Minister, and these are the different 16 departments which come under the National One Health Mission, that include every department which you must be knowing: DBT, ICR, DSD, and so on. So all these departments now committed to work together, and so that we are not going to duplicate, we should not do the same kind of effort in fragmented ways. And these are the some 6 main objective of this mission. The first, we should have a research and development to make the medical countermeasure. We should develop early warning signals. We should be ready for any outbreak or pandemic, and we should able to engage with different stakeholders on every matter in a way that India should become a better country. So the first action which was done by after establishment of National One Health Mission to create a laboratory network of BSL-3 facility coming under DBT, ICR, DSD, and also an ICMR and so on. And under this we have now 23 laboratory, and
a disease prioritization exercise was done with the help of all the stakeholder, and these laboratories should able to detect 39 human and 15 animal pathogen whenever there is an emergency or outbreak happening. But this is not enough. We need to train the staff, and so multiple training is going across the country, different with using different specialization, their expertise, and we are training a large number of human resource so they are ready to work in these laboratory. Did you ever heard about the mock drills? Do you know what is a mock drill? So before board exam, we do pre-board, right? Did you hear that? Pre-board exams. So for these laboratory, we also have a mock drill. That's a pre-board exam where you do a mock exercise so that you are ready when the real emergency come, like your board exams. So we did the country's first national mock drill, which happened last year, and this year also again we did a mock drill, which is done at national level, and we find out what is missing in the system and we are trying to improve it. Then in the past, we mostly detected dengue separately, chikungunya separately, and we keep running behind the pathogen. But now we understand that we have to do disease surveillance as per syndromes. So now we have divided diagnosis in syndrome wise, is called acute encephalitis, respiratory, acute fever, diarrhea, and also, and accordingly syndrome we have now developed diagnostic algorithm, and we are working, and these diagnostic algorithm helping us to find out more systematic data, and with the data driven diagnostic algorithm further will be enhancing our detection and surveillance system. The first and most important is the respiratory surveillance, which is done in 73 sites across the country, where we are able to detect weekly different pathogens which is circulating and which is emerging as well. The next is acute encephalitis syndrome, which is again done multiple sites. And the beautiful part of this acute encephalitis syndrome that we are able to increase our diagnosis using NGS by 45%. So if you do any diagnosis by different using all the tools, you can only detect up to 20%. But using the NGS enabled tools, now we are able to detect highly, and we are now able to develop algorithm using these data that what pathogens are missing. So this is one of example where we use the negative samples of AS cases, and we could detect many pathogen which was missing using the existing diagnostic algorithm. And they are not only viruses. That's what I want to tell. When we talk about the One Health, it's not about virus or bacteria. It's about all pathogen and how co-infection, how the virus bacteria co-infection is affecting the health of animal or human. So altogether how it is impacting the health environment. And when we are doing it, we also could able to do the AMR surveillance, because you are treating these animals, humans, while we are talking about disease and pathogens. The next surveillance program is in slaughterhouse. We being a veterinary, you know what it means, slaughterhouse. And when you slaughter the animals, there are so many workers, and there are possibility that when they are slaughtering these animals, there are ticks. Maybe the tick can bite to these animals, and then they can get some infection. So the health of any human is observed, and the blood of slaughterhouse samples are taken, and then try to triangulate to see if there is any outbreak happening. And this is happening in coordination with different sectors, and the initial study is leading to improve further how we can enhance the surveillance in our country. The second program, which is called we are working on the human bird sanctuaries, where we can monitor the interface between human and birds. You must be knowing that there are a lot of birds which keep traveling from one country to another, and there is a possibility that they can through the parasite and through the infection, they can spread disease from one country to another. And by such a human interface study, you can detect the ongoing outbreaks, and also if there is any emergence of disease. Again, it's a multi stakeholders study, and we are working. So when we work together in such kind of very complex project, these are not easy projects, I must tell you, because there is no algorithm, there is no protocol, there is no SOP. So we are building us itself a lot of protocols which was not done in the past. Then there is a next surveillance program which is initiated by TICS Bangalore and ICMR, which include a large network of VRDL laboratory, where we are working on wastewater and AMR surveillance. And I think this data will be very helpful to know more on that. India has never done air surveillance, which was missing. And now we are also even doing the flight surveillance, where we will collect the waste from the flights, and then coming from the different foreign countries, and those data would able to help us to do the early prediction of different diseases arriving in India. A very good example of very quick performance which we did with the mpox. As soon as in 2022 mpox emergency was declared, 34 laboratory was prepared. They are testing very meticulously. Genome sequencing is happening very quickly, and this data help us to know about which clade is coming. Most of the cases are coming from different country. If in India we do not have a lot of monkeypox cases, and some of cases which we observe mostly coming from one person to another, happening with the coming from some other country only. So we also recorded clade 1b cases, which is the most deadly pathogenic strain, and it can kill up to 8% of pediatric cases. So this monitoring itself represent that how alert our system now in compared in the past. We have also developed IVD diagnostic guidelines and release along with the CDSO, and that will help in validation of new emerging technologies and kits which we are working. As we all know that during the pandemic we had INSACOG network, where we were sequencing SARS coronavirus, but as soon as the COVID slowed down, we are not having any interest, or we don't know what to do with this infrastructure. So now there is a metagenomic hub created, and we will try to sequence all One Health aspect samples. It means that human, animal, and also the environment sample. So we can link a story of outbreaks, or if something happening and it is not in our attention. So these surveillance data will be very important as well. The most important, and you all has to understand the biosafety and biosecurity. When you go to media, when you write something, or when you react to something, somebody is watching you, somebody is looking at you, what you are writing, and who are you. And you must be knowing a lot of story where people are trolled, people are being taken because they have commented something badly on somebody. So that's called that is kind of time we are living, where everything is being monitored. And when we work on any pathogen, when we work on in any field, it means that you are part of this system. And recently when the Op Sindoor happened, you all aware that how different forces work together to safeguard our country. Similarly, the all three different sectors, veterinary, environment, and human health, together, by assuring the biosafety and security of country, we have to work safely on these pathogens. So that's another angle of One Health, which is very important and has to be understood. uh, ICMR has started a MedTech Mitra program, where we are nurturing not only the manufacturing of kit, but also validation, helping them in different ways, so that these kits can be taken to CDSO. And actually these efforts, there is a particular department, they are funding lot of projects. So if you have any interest in a new technology, if you have any idea, please explore the ICMR web page. There is a lot of opportunity to funding. If you have any great idea in One Health, you can reach out to our team. We will able to help you to develop your project. We will help you to write your project if you are a naive young researcher. In fact, this year there was a call for the first thought. If you have an idea which nobody had thought, we will give funding for that also. And if you need help to develop those projects, we are ready to help you to write that project. Why I'm speaking? Because this is a young audience. I want you to know that you are not alone. There are people who are thinking about you. They want you to invest in the research. They want you to invest in innovations. And I think that's how we will become part of this ecosystem where we talk about One Health. So under this program, there are certain products which we are able to get successfully successful on the path of making the KFD vaccine Nipah monoclonals in collaboration with Zydus and THSTI. We are also working on Zika vaccine, and also Dr. Naveen must have mentioned about H5N1 vaccine. So these are the four very important viral pathogen, but we have funded a large number of One Health projects where many institutes are working on developing new technologies, making vaccines, monoclonals, antivirals, and so on. So if you also have any ideas, you can ask, and we are there to help you for doing research. Even if you are working in a PZ, there is a funding opportunity for that also. So please come forward, take interest, and there is a lot of fund available. That's what message I would like to give you in one field, not only in the One Health but also in the R&D. And you can make any new product. If you have any idea of doing a small innovation, you can definitely do it. There is a platform available for it. So with this, I would like to thank the opportunity given to me from the university to come and speak and interact with you. But I would like to also thank the leadership from PSA office, Shri Ajay Suji, Dr. Rajiv Behal sir, and my colleagues from NIUH, from ESL4, from NIV, and Dr. Naveen, who to be with him I work, and all the senior members who are here today listen my presentation. Thank you, and I wish you all the best, all the young youth who are here. I'm sure that whoever win, they will get award in Bharat Mandapam. But those who don't win, don't get disappointed. You earn something, you learn something in this process while you are here today. Thank you so much.
Thank you, ma'am. Any questions? Yeah.
So I've seen 240 samples and 45% positivity even with NGS. Now, what do you think about the other cases where even NGS is negative? So do you think it is going to be autoimmune kind of a thing or something else? Because NGS is also not able to find out any bugs in that.
So this is just a preliminary data. We are working more, and there is another aspect to that work is the novel pathogen discovery which we need. So I am assuming that it will increase up to 60%. But yes, but we have to always remember that everything is not because of pathogen. Many time AS can happen because of other thing also. So not every time when AS cases arrive in a hospital because of just viral, bacterial or fungal pathogen, it could be anything. For example, recently we learned that amoeba can be so fatal, and in Kerala that can cause meningitis and so on, which nobody noticed so many years. So there are a lot to learn, and unless we have a very good surveillance system, we won't know what is happening to our population, why people dying, and what is the reason for death. So yes, we need to do a lot more, and I think we need more latest technology. All the technology. The sad part of in India that we use all the machines made in US, China, Singapore. We don't make any equipment, so we have to do a lot of innovation to make the equipment. Second, we also need to upgrade the science. The new knowledge should be generated. Why we should able to detect everything and in best possible way. So now everybody talking in the latest technology, CRISPR, but why we can't have our own new technologies? So that's what these young youths people has to be motivated. They have to invest in it, and I think that's the future lie in their hand. Thank you.
Any more questions?
Otherwise, I'll ask the question. Okay, thank you. If no question, I'm available here for some more time. Otherwise, you can send email to me. Our team is here in the Nagpur. You can reach out to them. If you have any questions, suggestion, you want to get funding, we will be very happy to support you. You want to do a small project, we will be able to help you. So our purpose to be here, our purpose to here to support you, to bring a new generation of scientists who can be doing better than us what we did. Thank you.
Thank you very much, ma'am, for sharing your valuable knowledge and experience with all of us. I would like to call upon Dr. Shirish Upadhyay sir to felicitate Dr. Pragya Yadav, ma'am.
Now I welcome Dr. Nagendra Har Egde sir, a distinguished epidemiologist advancing India's disease surveillance intelligence. Dr. Hegde sir represents the National Institute of Animal Biotechnology, Hyderabad, where he works extensively on disease modeling, genomic surveillance, host pathogen interactions, and predictive epidemiology. His contributions have helped build India's integrated surveillance systems, enabling early detection and forecasting of emerging diseases.
First of all, let me thank MAFSU and NIOH for inviting me here. Dr. Upadhyay, called me and said, "You have to be here, sir. You have to come for this occasion." So I couldn't deny. So here I am, and I am the last batsman. So and I'm going to talk about vaccines and phages to combat AMR. And I have to tell you a couple of stories before that. I don't know how many of you were at the quiz and were surprised by some of my questions that I was asking about bees and elephants and whatnot. So I can tell you a personal this thing. My father, who is 85 years old, Ayurvedic physician who still practices at the village. This year he called me twice. Once was to tell how much rain was there this year. He said, up until this year we were counting in centimeters. This year it's in meters. So this is also a One Health issue. The second thing he was telling was, oh, we are hearing that, there's a stream running by and they are going to build a dam to take the water to a city that is 200 kilometers away. Now, I'm scratching my head. Why is a dam being built to take water to somewhere 200 kilometers away? I understand there is human habitation, we need water for everybody and things like that. But in doing so, my home is going to be submerged. Plenty of Western Ghat trees are going to be submerged. Plenty of birds are going to lose their homes. So just wanted to give a flavor of what goes on at those levels that we also need to think about. That is also One Health issue. So my foray into AMR is very, very young. I am not a microbiologist. I am not a bacteriophage person. I recently got into this because of certain coincidental things. I am a mammalian virologist, but now ending up with a bacterial virologist. So bugs, uh, pardon me, this talk is mostly for the people who are at the back, not for the people who are sitting in the front row, because I wanted to speak more for the students than the experts. So you all know bugs are part of our life. They outnumber us. If you actually count the number of cells versus number of bacteria, so it's actually the 10:1. It should be the reverse ratio, but now it's been pared down to 3:1. And 1 to 3% of your body weight is because of microbes. Okay, so we carry so many microbes, and they also cause diseases. As you can see in several countries who are underdeveloped, they cause a lot of diseases, including simple diarrhea and things like that. I can give you one example, the rotavirus vaccine that was developed in India. So in the West, the rotavirus vaccine is used for reducing economic costs, meaning hospitalization costs. Children don't die. So up until we had the rotavirus vaccine in our immunization program, about 50,000 kids were dying of rotavirus vaccine. So different scenarios. Okay, so these exist in underdeveloped countries, also in some developed countries. So stopping antibiotics is not an option. As of now, we can't stop antibiotics. They are a very essential medicine that we require. Okay, but then we also have this issue with AMR because we use antibiotics at will. There's also a huge impact on the economy. Right now, there are about 7 lakh people who are dying out of AMR. And by 2050, this is projected to be 10 million. And it's at a huge cost to the economy. About 3 to 4% of the gross domestic product is going to be hit. And the biggest hit is going to be on the low and middle income countries. This is the curve of antibiotic discovery. As you can see, started in 1940s. There was a peak somewhere between 55 and 70, 75, 20 years of glory, and then a precipitous decrease. And now it's almost down to zero. And no new classes have been identified for the last more than 3 decades. And they're only used for short term. New drugs are restricted to mostly resistant microbes, and the libraries from which they test all these compounds have exhausted, and resistance has been discovered soon after a new antibiotic is administered. So much so that the British Medical Journal put out this pipeline. So the antibiotic pipeline has basically dried up. And one of the major drivers is the long lead times, which are not attractive to the companies. So for so those of you who don't know how drug discovery happens, if you start with 10,000 to 20,000 compounds, you might end up with one blockbuster. And if you think that why are these new medicines so expensive? It's because it takes 15, 20 years for a company to develop this medicine, and they have to recover the cost. So that is where the company is going, and the break even is several years for antibiotics, mainly because as soon as they introduce, they see resistance. So they can't recover much. And that's one of the reasons why companies are really not interested in new antibiotic discovery. And if you look at how the antimicrobial resistance occurred, and you can look at all these red bars ending with the X at the end. So within a span of anywhere between 2 to 5, 8, 10 years of introduction of any of these antibiotics, you would have seen resistance. It's not that, that resistance arose because of the antibiotic. It was already there, probably was amplified. Okay. So, but you have to think the other way. These dates are only about the discovery of the resistance, but not the existence. Did they exist before? This is a chicken and egg question. Which came before? Did AMR come first, or did the bugs come first? Okay. So there have been some studies. Somebody did some studies with the 30,000-year-old permafrost samples, and they detected AMR genes to tetracycline and glycopeptides, which means bacteria harbor them. But by overusing antibiotics, we are amplifying such bacteria. Okay, AMR existed before, it's been amplified by us. And the flow of AMR is very complex. So you have bacteria that are there in the environment, bacteria that are there in animals, bacteria that are there in humans, and they crisscross everywhere. And there is transfer of genes. So it's very complex. If you can see these arrows flowing, they're going from everywhere, including effluents where industries are synthesizing antibiotics and putting it into the water. There also you will see increase in AMR. In fact, in food animal production, it's been hypothesized that this is one of the largest contributors of AMR. In fact, it's true in China and certain Western countries where there is large scale production of pigs and cattle and things like that. So there has been a hypothesis that livestock production is contributing to AMR quite a bit. And if you look at this graph, I want you to focus on the topmost right-hand corner, where both the evidence that a factor contributes as well as the relative contribution are shown in big two blobs, and both of them are like, one is a human antimicrobial misuse, and the second is an animal antimicrobial misuse. Either way, it's the humans who are doing it. Okay, so we are in fact responsible for this crisis. So we need to somehow reduce it or use alternative to antibiotics. So what are the alternatives? I've just listed some here. Starts from vaccines, immunotherapeutics, antibody conjugates, CRISPR-Cas editors, probiotics, microbial therapies, phage therapy, fecal microbial transplant, nanobiotics, stem cell AMPs. So I'm not going to talk about all of these, but I'll focus on two things, which is vaccines and phage therapy. And I am not going to give a whole lot of scientific data. I just want to brush up on the subject to give you more insight about how these things contribute to mitigation of AMR. Now you might think AMR is mostly because the antibiotic is resistant and it is carrying certain genes. How would a vaccine mitigate AMR? If you have a vaccine against a bacterium, how would it mitigate AMR? So I'll give you a scenario here. If you look at the top, a single individual gets infected. You use a first line antibiotics, and then either the infection gets cleared, or in the second case, you get an emergence of AMR resistant bacterium. And then when that person is introduced into the population, that resistant bacterium actually spreads. okay, because the susceptible one is already killed. Now, this is what typically happens. What happens if you give a vaccine? Okay, if you give a vaccine, you're basically preventing, whether it's resistant or susceptible, it doesn't matter. You are preventing that bacterium to spread among the population. So this is how a vaccine can actually decrease the spread of the bug and help in the mitigation of AMR. Now, the second half of that graph that I showed you earlier, the top is what I have already shown where the resistance occurs. If you look at the bottom, resistance to vaccine is unheard of. Okay, so there are one or two X's here. One of them is pertussis, other is meningococcal. Both are bacteria, and it's because of difference in strains that the AMR has happened. It is not because the vaccine is ineffective. Okay, vaccines never fail. So it's better actually to go for preventive rather than going for antibiotics. So is there evidence? I'll go with two examples. One is Streptococcus pneumoniae. This is in humans, causes a very common respiratory tract infection. There are more than about 100 serotypes that are there. And the other that also causes respiratory tract infections is Haemophilus influenzae. For both of which vaccines are available. One is called PCV, the other is called Hib vaccine. So here is one graph I'm putting together. This is the cases of invasive pneumococcal disease, so-called IPD. On the y-axis is cases per 1 lakh population, and on the x-axis is the year. And the lines, the vertical lines show when was the PCV7 introduced and when was the PCV13 introduced. So you can see in the red line a precipitous decrease in the cases of IPD after the introduction of PCV7. The other lines are actually the strains that were not there in PCV7. So they are pretty much constant. This clearly shows that cases of IPD and hence AMR is reduced by using this vaccine. The second is Haemophilus influenzae. So this again shows this orange graph where there is a precipitous decrease. And of course, there had to be a catch-up campaign when there was a blip. So that again reduced the incidence further. So clearly indicating that this is reducing the number of these incidences and hence dissemination through the population. So I'm going to spend a little bit of time on this slide telling you what are the different ways a vaccine can help in reducing AMR. Number one, it can reduce the incidence of disease and infection, which I have shown with a couple of graphs. And then because it reduces the population carriage, it reduces the colonization in vaccinees. And because it is reduced in the people who are vaccinated, it reduces the carriage in in-contact individuals. So if you have a family and I'm vaccinated, of course, I can't spread it to other people in the family. It reduces OPD visits, reduces hospitalization in ICU and exposure, reduces prescription, because if you have less cases, there will be less prescriptions, which means less use of antibiotics, less antimicrobial use, less medical expenses, less school and office absenteeism, less transmission, limiting exposure to other people, and less resistance overall, as I have already shown, and therefore AMR transmission. And the protection is much longer than the treatment. Remember, your antibiotic treatment gives you protection for about 3 to 5 days, maybe a week, 10 days, but this will give a longer protection. Okay, and so if you actually look at, if you try to calculate the economics of reduction of all these things compounded together, nobody has done this in the world. You can't really quantitate certain things. For example, absenting from school, how do you quantitate? How do you quantitate an economic benefit? I tried to look for literature for this, and I found out some vague abstract from in Canada where they said if you spend $80 billion for this, you will save 20 billion. So that is the extent of savings, but nobody has actually put in mathematical models, etc., for the economic benefit. What do we target? Some of you may have heard about these escape pathogens. These are the most common, and these are Escherichia coli, Staphylococcus aureus, Klebsiella pneumoniae, Acinetobacter baumannii, of course, mostly human, Pseudomonas aeruginosa, and Enterococcus. So except Acinetobacter, all the others are shared between humans and animals. Even in animals, we are finding Acinetobacter these days. Okay, so So I talked about vaccines. The other thing I wanted to talk about with, couple of slides maybe with bacteriophages in mitigating AMR. I wanted to give a little bit of history of bacteriophages. Bacteriophages are also your microbiome. Wherever there are bacteria, there are bacteriophages. They are very widely distributed, highly ubiquitous, and they outnumber every other organism on earth. And it's estimated that the number of bacteriophages in number of particles is 10 to the power of 31 on earth. Okay, they are the second largest biomass after prokaryotes, and 70% of the marine bacteria are infected with bacteriophages. And in the marine surface microbial mats have 9 to the power of 10 to the 8 virions per ml, and they are very important in controlling the flux of bacteria. Why I say flux? If you have more bacteria, bacteriophages infect, and the bacterial number comes down. Now the bacteriophages cannot survive because they have to infect their host. So once the number comes down, the bacteriophages cannot infect that much, the bacteria will go up. Again, the bacteriophage will come up and reduce the numbers. So this flux in biomass, especially in oceans, is very important. Okay, so they play a very important role in controlling bacteria. And I've given a graph here about bacteriophage therapy. It started very early, late in the 19th century, early 20th century. Already the Russians and these people were using bacteriophages, but then started the antibiotic boom. The bacteriophages are actually quite specific. If you have a bacteriophage against E. coli, it will only work against E. coli. But an antibiotic, on the other hand, will work against Staph aureus, E. coli, whatever else. So they are broad spectrum. So people started using antibiotics because they are broad spectrum and simply you can give it without thinking. So, but then started the AMR problem, and that is where bacteriophages are now becoming important. It may take a few years before they become mainstream, or they may not become mainstream. I don't know. It may take time, but they are here to stay. So what are the differences? If you look at antibiotics, antibiotics target pathogens plus beneficial bacteria. If you have heard, or if you have gone to a doctor, every doctor prescribes antibiotics. Along with that, they'll give a probiotic. Okay, many times they give you a probiotic or an antacid or some such thing. That is mainly because they want to keep your healthy gut microbiota alive. Okay, so they also kill beneficial bacteria, but bacteriophages are specific. Okay, you can actually target specific pathogenic strains. Pharmacology, antibiotics are metabolized and eliminated. Bacteriophages, as I said, they depend on their host, or they only replicate where the bugs exist. Side effects, antibiotics do have side effects, intestinal disorders because of microbial dysfunction, allergies, secondary infections sometimes. Whereas bacteriophages, they're not serious. So far, we have not seen any adverse events, shown in humans. Cost and time taking to produce, develop antibiotics and expensive. Bacteriophages are very rapid. Within a week, you can get some bacteriophages, and it's cost effective. And they're also effective against biofilm, which many of the antibiotics are not. Okay, so bacteriophages can be used for many things, including pest control. So you can use it as spray for vegetables and fruits, which is being used, or dental or medicine, and even in methane control in animals. So we have done some work with bacteriophages. I will not go through the details of how we did and all that. What we have done is we have taken 250 samples from various places, and we have isolated 65 bacteriophages so far. 37 of them specific to Escherichia coli, 22 to Enterococcus fecum and faecium, fecalis and faecium, and 6 to Staphylococcus aureus. And we have characterized them through a battery of tests, including temperature stability, pH stability, morphology, RFLP, genome sequencing, cytotoxicity, shelf life, everything we have tested. And we have also tested whether they work in milk. So hopefully one day we can also use this in combination as slightly as a broad spectrum. Beyond phages, also there are certain things. So bacteriophages have certain enzymes that they use while entering and exiting the cell, exiting the bacterial cell. So these are called endolysins and ectolysins. And endolysins and ectolysins can actually be used as therapeutic agents, because with bacteriophages you have high specificity, but with the lysins you don't. So you can actually use them as slightly as broad spectrum. So endolysins can be various kinds. You can have peptidoglycan hydrolysis. You can have biofilm disrupting enzymes, autolysins, bacteriocins, or other lysozymes. So these can all be used for therapeutic purposes when you express them as proteins. So this is actually the carriage of different enzymes that we did with our own bacteriophages. We sequenced about 18 bacteriophages. So this is just a list of enzymes that we found through genome sequencing in E. coli phages. So I won't go through the details, but both bacteriophages and in particularly endolysins have been used in phase 2 and phase 3 trials. In fact, this P128 lysine that is in the third row of the top table is from an Indian company called Gangajan. So they are already in they are going for phase 3 for nasal carriage of Staph aureus. So, but on the other hand, bacteriophages are commonly being used for keeping the shelf life of vegetables and meat. So that is shown in the bottom. So AMR mitigation requires multi-pronged strategies, and one is not enough. So there is plenty of room for anybody to work on alternatives, including plant-derived antimicrobials. And I've only given you a glimpse of what vaccines phages can do. And definitely we need to work together in order to beat this beast. Thank you.
Any questions?
Yeah.
Bacteriophage as well. So what's your thought on that?
So there are a couple of things people are trying. One is people start with thousands of phages against a particular bacterial species, and then they try to identify those that are broader spectrum. For example, with E. coli, we have 35 phages, of which one of the phages has consistently lysed more than 60% of the strains that we have tested. With Staph aureus, we have 6 phages that are over 90% of the strains are being targeted. So people are trying to identify those that can work at broad spectrum, or you can use a combination. So people are also trying a combination of the 4 or 5 different bacteriophages that can be used at a time. The third alternative is you can actually use bacteriophages first, and then 3, 4, 6 hours later hit with antibiotics. So that is the other thing that people are also trying. And at those cases, you can actually do subtherapeutic doses of antibiotics after treatment with therapy, phage therapy.
So if I am correct, so the bacteriophage as such is not allowed to use as a therapy. Only endolysin, right?
As of now, no. Bacteriophages are only allowed for what we call as compassionate use, where there are cases where people have completely failed to treat somebody. There is actually a case in India also. That fellow went through a serious nephritis due to Klebsiella pneumoniae, and nothing worked. So he was suffering, and he had to get phages from Belgium because it's not allowed in India. And through the Geneva Convention, they got a consortium of phages from Belgium, and he was treated successfully. And he goes around giving talks, I'm alive because of bacteriophages. So it is, as of now, allowed only on compassionate grounds. It will take quite a bit of time before it becomes a drug, because drug by definition has to be very uniform. You have to define a drug substance, drug product, all the process, how much input goes in, how much output goes in, certain processes. Those are a little difficult to define for bacteriophages. The closest we have are viral vaccines, but even they are not the same as bacteriophages. So I think even the regulators need to pour their heads over this and figure out how do we allow this. And ultimately, whether this will become a product, that is $1 million question. Because if I am a company X, and I want to develop bacteriophage, let's say I produce 1 kg of bacteriophage that works on only E. coli, and I see only 2 cases per week. Is it worthwhile for me to do it? Probably not. So it's a combination of economics versus, what will happen.
Okay, thank you. Thank you very much.
As a preventive than the therapeutic, and it is contributing more for AMR. So can we use these bacteriophages as a preventive therapy or instead of antimicrobial growth promoter, can we use this as a preventive therapy?
Definitely, yes. And especially when you're doing large scale production, especially for example, poultry. I'm talking about poultry because in poultry, there are only very few lines of birds that are being used. So very likely their gut microbiota, because of the feed composition and everything, will be very similar. And you're not going to target too many variants. So I think there's a definite chance for use in poultry. And I know that it is already being used.
It is used.
Yeah, thanks for the excellent talks. A lot of good information about the One Health angle. So, don't you think we have to put the regulatory people on the board right from the beginning till we are starting working with the research? Because if we bring them later after 7 years, 8 years, we have to start from their angle and all these things. Because we faced a lot of problem with the FMT also. In India, we started work with Indo-UK collaboration. So we found a very difficulty in giving a CDF treatment and all these things. So don't you think we have to do something so that we can bring them on the board very early so that they will also try to give their things from the production perspective or approval perspective?
I completely agree. In fact, ICMR and the phage group within India is already working with the regulators, representing and things like that. The biggest problem is in drug development, the standard is clinical trial. So phages have not reached a position where we can do clinical trial, because if there is a single patient, how do I do a clinical trial? And how many patients do I need to include, and how many days do I need to wait for that particular condition to appear? For example, osteomyelitis. How many cases do we see, per month? And how many control cases do we do? So, and is the depth of the problem or the seriousness of the osteomyelitis the same in all those cases? So how do we control all these things? These are a problem that is right now we are trying to grapple with. And one of the ways people are trying is you build a number of cases over years and then try to write systematic reviews, or we develop methodologies even for these single cases so that everybody uses similar cases or similar procedures for doing these studies, so that when we do the systematic studies we filter out all those confounding variables. So it will take time, both from the side of the clinicians and from the side of the regulators. We need to work together.
So I just want to add something. So we have many examples wherein the disease incidence or number of cases we are getting very few. For example, Chandipuras. So last year we got maybe 300 cases, but before that 4, 5, 6 years there were no cases. KFD we are consistently getting some cases. So in that case, if you want to involve the industry, they are not willing to, because there is no market. So what we are doing in at least 3 of the cases, for example, KFD, although one of the veterinary institute, they were making the vaccines and now some issues. So big industry partners are not coming. So what we are doing is the ICMR is facilitating the industry. So this vaccine is being, in collaboration with the Indian Institute of Immunologicals. So we are doing that. Similarly, in case of the Chandipura, so we have to do the same because industry will not come. So sometimes what happens, you did a very good research and did not involve industry. I fully agree with you. So like what happens with us, So as per the WOAH guidelines, when I was working on LSD, so we did all the trials, experimental trials, field trials, whatever that is required. The data were published in very good journals. Then eventually, we transferred the technology, and then uh, the drug controller said that the vaccine that is being used in the trials, it was not GMP. So you have to do everything with GMP. Yes. Yeah. So that's why we have a BSL-3 lab, BSL-4 lab, and is as par, but it's not GMP. So the trial means everything should be GMP. That's why we have to involve the industry. So that's the limitation of both the sides. Thank you.
sepsis and all these things is quite high even observed. So there are 10 or 12 cases where MDR is quite reported, and colleagues of mine and all those things who are working, they switch over to second line of treatment. So I think this is the bedside experience I'm putting in this forum. So this is exactly what we feel when patients, we're not able to treat patients, right, in spite of everything. So this is what I'm putting.
Yeah, as of now, phage therapy is a doctor's choice. So that's, and I also, if I can add to Dr. Naveen's this thing. So if you see how it works in the West, a lot of money is actually given to industry to do things, and in return, industry puts a lot of money for R&D. Here, it's a little bit different because our priorities are entirely different. We have much more pressing problems to deal with, and we can't really put that much money on either side for these kind of things.
Yeah, so actually, to break that, there is now at least a small amount is given to industry for each R&D. So that's a new pattern which government of India is trying, and ICMR is also trying to do that, because otherwise, why they should invest in it? They are like, and many time actually they are even doing 50% is still investment in it. Then only you can get some little answer. I have a small questions to you regarding the bacteriophages.
Yes.
I met with a VHU researcher who are very passionate with the using bacteriophage for burn injury cases. So how feasible is those kind of treatment protocols in India? And I think he keep driving this agenda to many people, but somehow it didn't work.
Yeah, I
Is
think you're referring to Dr. Gopal Nath.
yes,
So
yes,
he has done quite a bit of work on, as I said, osteomyelitis, burn cases, and
yes.
serious cases of fractures where bones were exposed and things like that. So there, the decision, as I said, is made by the physician. The physician ultimately decides whether nothing is working, so I need to do something, I need to do something for the patient. Then they go to Dr. Gopal Nath and they say, can you help us? And in such cases, these are individual cases, not case controlled. So these are individual cases on compassionate grounds. So these are driven by doctors, and that is where right now the much of the data is being developed. And he has wonderful, clinical pictures where in one or two cases he also has done controls where people have used antibiotics versus for burns you can do that, but for osteomyelitis you can't because there isn't much you can do for osteomyelitis. So
Okay, thank you. So you are aware of it.
thank you. I think there's one question there if we can.
So in case of ocean cycle, there is a balance between virus and bacteria. So if we continuously keep on using phage therapy in human, can this balance shift to similar to ocean in human?
Very good question. But as I said, the moment you use, let's say I have some infection and I use bacteriophage and it somehow infects my gut E. coli. Okay. So there is some amount of disturbance, but once that E. coli number goes down, the phage cannot survive anymore because it doesn't have its host. That's why I was talking about this flux waning and waxing. So once its host goes down, it cannot survive. Again, the host has to come back. And in many times when the host comes back, it has a different receptor. It's no longer accessible to that particular phage. So this is how it usually works. It's a balance at the biological level.
Thank you.
Thank you.
Thank you very much, sir, for your deeply informative and thought-provoking session. I would like to call Dr. Kurkure, sir, to felicitate Dr. Hegde, sir. We express our heartfelt gratitude to all our esteemed speakers for enlightening us with their tremendous expertise. With this, we conclude the technical sessions. A round of applause for all. Thank you.
We now begin one of the most exciting and high energy event of the day, the One Health Quiz competition. All teams are requested to take their respective seats. Our quiz master will brief you about rules before we start. This quiz will challenge your understanding of zoonosis, public health, environment, epidemiology, One Health policies, and global health intelligence. We wish all the teams best. May knowledge guide you, the team strengthen you. Let the competition begin.
Hello. Hello. Check. Check. Check this. 21. 21.
Participants, please come to the stage. We have 8 teams. First team from Kamdenu University, Gandhinagar. Second team, Rajuas, Bikaner. Third team from Bhubneshwar. Fourth team from Kaksu, Bidar. Fifth team from Rajuas, Jaipur. Sixth team from Durg. Seventh team from Hyderabad. And eighth team, our own team, Mafsu.
Robust.
Please check the letters written on your name like A, B,
Hello.
C, D team. Please remember you won't be called by your university name. You will be named A, B, C, D.
Please take your number.
28.
Hello, everybody. Good evening. Am I audible? So my name is Padmanabhan Pillai. I'll be the quiz master. I'll be assisted by Ms. Anushka Nag. I belong to an organization called Nagpur Quiz Club. So we started in 2011, and over the years we have conducted close to 150 quizzes in many parts of India. The first thing I'd like to tell you is we are not here to test your knowledge. It doesn't matter whatever you know or you don't know. The idea about quizzing is everybody who's here to participate or even to sit in the audience, when you step out of the room, you know slightly a bit more than what you knew at the time of entering the room. That, my dear friends, is the beauty of quizzing. So it doesn't matter whether you know the answer to a particular question or not. We are here to learn some new things, and quizzing is fun. So let's keep the fun aspect alive. Everybody here is ready. Let's have a quick round of introduction. Team A, your name and your college or institute, please.
Myself, Popalya Kashyap. I am from Junagadh, Kamden University, Gujarat.
My
A big round of applause for Team A from Gandhinagar, please. Kamdhenu University. Team B.
My name is Paransha Adhikari and I'm from CVS Bikaner.
My name is Shreya Singh. I'm from CVS Bikaner.
Round of applause from Team B, Bikaner. Team C.
Myself, Asutosh Panirahi from College of Veterinary Science and Animal Husbandry, OUAT.
कुछ भी नहीं समझ में आया मुझे. Once again, please.
Myself, Asutosh Panirahi from College of Veterinary Science and Animal Husbandry, OUAT.
Okay.
Myself, Vedadipta Sahu. I am from CBS OUAT.
Team C.
Myself, Pallavi from Kofsu Bidar, Karnataka.
I'm Abhijit Thomas from KVFSU, Bidar, Karnataka.
Team D from Bidar. Your team E. Team E from Jaipur. Team F.
I am Dr. Rizwan Khan from College of Veterinary Science and Animal Husbandry, Durg.
My name is Rishabh Shande. I am from DSVCKV Durg.
Team F from Durg.
I'm Khushi. We are from PVNR TVU, Rajendranagar, Hyderabad.
I am Manaswini. We are from PVNR TVU, Hyderabad.
Team G from Hyderabad.
Myself, Sneha Subhash Kore, Mapsu Nagpur.
Myself, Dr. Yogesh Sorat, Mumbai Veterinary College, Mapsu Nagpur.
The home team from Mafsu Nagpur. Let's have a quick look at the rules that we'll be following today. So overall, there will be 4 rounds. The rules will be told to you as and when we reach those particular rounds. Overall, there will be 42 questions spread across 4 rounds. Individual rounds for pounds applies unless explicitly told otherwise. Now, how many of you know what a pounce is? Nobody? Okay, I'll tell you. Everybody here has a sheet of paper, right? And also a pen. Okay, suppose the first question is a direct for team A, and the question is, what is the capital of India? So before team A is allowed to answer, I'll be asking whether any of the other teams want to attempt the answer. And suppose team C thinks that they know the answer, team C will just raise their hand and say pounds. Before team A is allowed to answer, I'll go to team C and they'll have to write their answer on the sheet of paper provided to you. I will not disclose whether the answer is correct or not at that point. And if no other team is pouncing, I'll say pounce is now closed and then team A will be allowed to answer. Suppose team A says Chennai. The question will pass on to team B. And team B suppose says Bidar. It will not pass to team C because you have already attempted on the pounce. It will go directly to team D. And team D answers Delhi correctly. I'll tell you the point pattern. Team A will get a 0. Team B will also get a 0. Team D will get 20 points. Suppose team C had given me the correct answer on pounds, I'll give them 20 points too. But had they written an incorrect answer on pounds, they'll get a negative 20. So in this case, both team A, B, and C, all of them are giving me an incorrect answer, but only team C is getting a negative because they are answering out of turn. Okay. Whenever you're answering on direct, there is no negative markings. Feel free to guess. But if you are pouncing, you're attempting a question that is not your own. So be sure you know the answer, else you'll be getting a negative point. Am I clear? Yes. Any doubts? Okay. Let's move on to the first round. Let's move on to round 1. In this round, there will be 8 questions. Pounds I have already explained to you. If any team gets the answer without hint, they'll get a plus 20. And in case you opt for a hint, the maximum points that you'll get is plus 10. So make your choice accordingly and pounces will vary accordingly. Clear? It'll be clearer when we reach the first question. The first question happens to be a direct to team A. Here is your question. X refers to a complex diagram that shows how different things are linked together. In One Health, the antimicrobial resistance X is a visual map that shows how resistance spreads across humans, animals, food, crops, and the environment. While X is a nickname used by researchers and is not an official term, the name X comes from how a person feels while looking at such a complicated diagram. Simply identify X. Everybody understood the question? We are talking about a diagram with multiple links and is a very complex diagram. Everything that needs to be there is present in the question. And you can identify some blocks over there. That is the answer. And if team A opts for a hint, certain letters from the answer will be revealed to you. But if you opt for a hint, the maximum point that you can score will be plus 10. Everybody understood the question? Okay. Now tell me, anybody wants to pounce? If you want to pounce, just raise your hand. Yes. What is your doubt? then it won't be the correct answer, right? Okay, I am closing the pounds in 3, 2, and 1. So pounds is now closed. Team A, do you want to answer for 20 or do you want a hint? Okay, if you can answer on the mic, then everybody will get to hear it.
Hint.
Okay, so team A opts for a hint. Now do you want to answer, team A? A complex diagram that shows how different things are linked together. There is a clue in the last line. While X is a nickname used by researchers and is not an official term, the name X comes from how a person feels while looking at such a complicated diagram. Okay, so team A, you're answering, or shall I pass on to team B?
Pass.
Okay, question passes to team B. Team B, can you pass on the mic, please, team A? Okay, team B, do you want to try or you want to pass? It's a very workable question. If you just focus on certain words, you will be able to reach out to the answer. You do not need to be technically sound to tell me the answer. Yes, team B. Okay, team B, I'll have to pass on to team C if you're not trying. Team C, it's your chance now. Team C is also passing. Team D, do you want to try? Or shall I pass? If you're trying, then maybe you can take a guess. I'm passing on to team E. Team E is also passing. Team F. Can you use the mic, please?
I have doubt.
Yes.
If answer given by me is wrong,
You will not get any negative. You will get a negative only if you pounce and give me an incorrect answer. You can feel free to guess.
confused row.
If you can answer on the mic, please.
Confused row.
I will give it to you. Plus 10 to your team F, right?
F.
Team F gets a plus 10. The answer indeed is confusogram. So whenever we look at this particular diagram, the first thing that happens to people is they get confused. So that's why it's called confusogram. Good attempt by team F, and team F gets a plus 10. No points to anybody else. Now everybody understood how pounds works and how hint works. Okay, so team A, you're done with your direct. The next question is a direct to team B. Team B, here is your question. One of the most common reasons for food-related illnesses in both humans and animals is a bacteria which causes an infection named blank osis. Despite of being very common, this infection can be avoided by using very simple steps like cooking the food properly, avoiding unpasteurized milk, washing hands, and handling animals with care. Although this bacteria was discovered by Theobald Smith in 1885, it was named after his superior, Dr. Daniel Blank. But for someone not aware of this bacteria, might just assume that it is named after a fish. What is the name of this bacteria? Plenty of clues over there. You just have to write down the answer on the sheet of paper provided to you. Team D is already bouncing. Okay. Any other team wants to bounce? Okay, you are team F, right? Team D and team F have bounced. Team G is also bouncing. Okay. Bouncing. So team D has bounced. Team F bouncing. Okay. So I'll tell you which all teams have bounced. Team D has pounced, Team E has pounced, Team F and Team G has pounced. So Team H hasn't pounced, neither has Team A. Every other team, Team C has also not pounced, neither has Team D. Okay. So pouncing time ends in 3, 2, and 1. So Team B, what is your answer? On the mic, please.
Hint?
What?
Can we get a hint?
Okay, so Team B wants a hint. It's much more obvious. Everybody sitting in the audience already know the answer. You can gauge it from their reaction. Yes, Team B, you want to try, or shall I pass it on to Team C? They seem to be very excited.
Is it salmonella?
Salmonella. And why will that sound like a fish? Is indeed the right answer. A big round of applause for all the teams that pounced and also to Team B. So Team B gets a plus 10. Team D gets a plus 20. Team E gets a plus 20. Team F gets a plus 20. And Team G gets a plus 20. Am I correct? So next question is a direct to Team C. Here is your direct question. Chlamydia is a bacterial disease that affects most living beings, including humans, birds, cattle, and wild animals. A strain of chlamydia is well known for the reduction of a particular animal in Australia. Transmitted through direct contact, this disease makes these animals blind, causes infertility, and gives them a urinal infection, which eventually makes them dehydrated and unable to find their food. In Queensland and New South Wales, the infection rates are suggested to be approximately 70%. However, very recently, the Australian government approved its first single dosage vaccine to fight against chlamydia, something that can reduce the infection rate by 65%. Which animal are we talking about? So we are talking about a particular disease called chlamydia, which posed a severe threat to a particular animal in Australia. I'm sure all of you know about chlamydia. But if you want to reach out to the answer, you just have to think about Australia and the animals that you can associate with Australia. And just check if the letters fit in the box. Direct to Team C. Anybody wants to pounce? Okay, team from Nagpur is pouncing. Team H. Okay, so Team H has pounced. Anybody else wants to pounce? Chlamydia. Animals of Australia. I'm sure whenever we mention about Australia, a number of animals come to our mind. Okay, Team B has also pounced. Team G is also pouncing. Okay. So B, G, and H have pounced. Direct for Team C. Any other team wants to pounce? I'm closing the pounce in 3, 2, and 1. Pounce is now closed. Team C, do you want to answer or you want a hint? Before saying hint, एक बार फिर पढ़ लो. You want to answer? I don't want to bully you. Hint भी ले सकते हो. You want to answer? Okay, so Team C is answering. Go ahead.
Sir, is this koala?
Koala. Is it found in Australia?
Yes, sir.
And chlamydia affects koala?
Yes, sir.
Plus 20 to Team C. And also to every other team that pounced, which is Team B, Team G, and Team H. Any other team pounced? Plus 20 to Team B, Team C, Team G, and Team H. So the next question is a direct to Team D. Here is your direct question, Team D. I'm stepping out so that all of you can see the questions properly. X, a zoonotic disease, although has its origin in Asia, it spread out to the entire world in no time thanks to transport cattle and ended up taking the lives of several domestic and wild animals due to its high mortality rate. Thanks to the efforts of British veterinary researcher Walter Plowright and others, a vaccine to fight X was developed, and by 2011, the Food and Agricultural Organization of the United Nations officially declared X to be eradicated completely, thereby becoming the only zoonotic disease to disappear entirely. Identify X. I'm sure everybody sitting here, you're aware of this name. It is the only zoonotic disease to disappear entirely. If you're not bouncing, your teachers might be throwing you out of the class. Direct to team D, every team is bouncing as expected. Complete silence from the audience, please. Okay. ठीक है. Okay. You're direct. Okay. 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 So every team has already got their plus 20. Go ahead, team D.
Rinderpest.
Rinderpest will fetch everybody a plus 20. Can anybody tell me which language is Rinderpest? For no points? No. Rinderpest is German. It's German for cattle plague. It's not zoonotic. Okay, and it's spread by cattle, right? Okay, I'm sorry. Okay, so plus 20 to everybody. Next question is a direct to team E. Team E, here is your direct question. In 2019, with $77 million export export, Denmark happened to be the biggest producer and exporter of the X fur, but the situation changed dramatically when COVID arrived. X's are amongst those animals that can be affected with coronavirus. The possibility of it acquiring the same from a human and creating an entirely new variant of COVID, which might also reduce the effectiveness of the vaccine, caused panic. And despite of public protests, the Danish government ordered the mass culling of over 1.5 lakh X's without any legal authority, thereby creating a huge controversy along with global criticism, especially after foreign experts from WHO stated that the risk from this new variant might have been minimal. What animal are we talking about? So Denmark was known for exporting the fur of the animal that you can see in the image. But this animal could also acquire COVID like humans, and there was a possibility discussed that it might create its own variant of COVID-19, and that resulted in Denmark government asking for killing of these animals. And it was criticized later because WHO claimed that if there was any chance of a new variant, the risk would have been very minimal. So it was something that created a lot of news. Or if you don't want to read the entire question, just identify what animal is there in the image. Direct for team E. Anybody wants to pounce? Team H. Okay. Team B is also bouncing. Okay. So two teams have pounced, B and H. Anybody else wants to pounce? Direct for team E. Okay. So team B has pounced, team D has pounced, and H has pounced. B, D, H have pounced, and 3, 2, 1, pounce is now closed. Direct to team E. Yes, team E. Can you identify the animal?
Sir, hint.
You want a hint. Okay. Team E wants a hint. Already 4 letter hai. Okay, you can see one N over there. Just identify the remaining 3 letters. No negatives for guesses.
Queen.
Hmm?
Queen.
Queen. Okay, team F.
Sir, mink.
Mink. That animal is mink. Just because you have N for the third position. Plus 10 to team F for answering mink correctly. And a big round of applause for all the teams that bounced. B gets a plus 20. D gets a plus 20. H gets a plus 20. F gets a plus 10. B, D, H get a plus 20 each and a plus 10 to team F. It's your direct now, team F. Here is your question. Right from its development in the 1950s, this pesticide X proved to be highly effective against insect pests, and it was soon used all over the globe. By the late 1990s, approximately 12,800 tons of X was manufactured annually. When the health hazard on humans and animals were noticed, the role played by X was easily brought to notice, because of which it was banned by many nations, including USA, in the early 2000s. However, India thwarted every attempt at the UN to ban the usage of X, as it also happened to be one of the largest manufacturers of X. Following an event in Kerala that is now called the X tragedy that resulted in severe health issues in humans and livestock, the Supreme Court had to step in and ban the manufacture and sale of X, which eventually came into effect in 2017, that is decades after the rest of the world. What is X? A highly effective pesticide, created a lot of news in Kerala, but it was something that was banned by US many years ago. It took India a lot of time to ban it, and by 2017 it was banned. Anybody wants to pounce? It's a direct for team F. Pouncing. Okay. Okay. Team H has pounced. Anybody else wants to pounce? Else I'll be closing the pounce in 3, 2, and 1. Pounce is now closed. Team F, you want to answer or you want a hint? Or you want to pass? Okay, so team F is passing on to team G.
Take a hint.
Okay, team G opts for a hint. Let's have a look. Yes, team G, you want to try? Okay, so team G is passing it on to team A. Only one team has pounced, that is team H. Team A, you want to try? Kamdhenu University, Gandhinagar. Passing on to team B. Team B is also not trying. Team C, you want to try? Okay. Team
Endosulfan.
Endosulfan. Are you sure? Plus 10 to team D for answering endosulfan correct. The endosulfan tragedy was something that created a lot of news nationally. And of course, everybody in Kerala knows about endosulfan, even if they are not into science. And a plus 20 to team H for answering it correctly on pounds. It's your direct now, team G. Here is your direct question. X was an Indian dairy engineer and social entrepreneur who led initiatives that contributed to the extensive increase in milk production in India, termed as the White Revolution. White Revolution, also called the Operation Flood, launched on 13 Jan 1970, was the world's largest dairy development program and a landmark project in India's agricultural history, which transformed India from a milk deficient nation into the world's largest milk producer. Just identify the gentleman about whom we are talking about. I'm checking who is not pouncing. Everybody is. Okay. So every team has pounced as I expected. Team G, don't tell me you want a hint. Okay, tell me what is the answer.
Vergus Korean.
Verghese Kurian is indeed correct. Plus 20 to everybody. Somebody said Verghese Courier. His name is not Verghese Courier. His story was turned into a movie. Can anybody tell me the name of that movie? Anybody from the audience? His story, which he co-wrote, was turned into a movie, and it was produced by 2 lakh farmers who gave like 2 rupees each, and they were all producers of the movie. The movie was called Manthan, directed by Shyam Benegal. Everybody must watch the movie. Last question of the round is a direct to team H. Here is your direct question. A 1980s epidemiologist called for a unified human veterinary approach, laying the foundations of modern One Health. Identify the scientist in question. Again, there are several scientists, but you have to fit in the name according to the blocks. Direct for team H. Okay. Not pausing.
Answer.
Okay. Okay, so team C, you haven't pounced. Okay, team C is not pouncing. मुझे एक बार answer दिखाइए. You're dead. Okay. So except for team C, every other team has pounced. Okay, so pounce time is now over. Team H.
Calvin Scobe.
Let's have a look at the answer. Answer, please. Excellent answer by team H. I'll tell you the point pattern. Team C gets nothing. Okay, but what's he alive in 1980s? Then he qualifies at 1980s. Okay, but he was alive in 1980s. So that's what I look forward to. Yeah, he has his book. Yeah, but he was alive. I'm not from this background, but the only thing I look forward to is, was the gentleman alive in the 1980s? Okay, thank you so much. So I'll tell you the point pattern. Team C gets nothing. Team A gets a negative 20. And team D gets a negative 20. Every other team gets a plus 20. Clear? So that is the end of this round. How do you find the quiz so far? Do you like the quiz? Thank you so much. Let's have a look at the scores. So at the end of the first round, team A is at 20 points. Round of applause for team A, please. Team B is at 110 points. Round of applause for Team B, please. Team C is at 60 points. Team D is at 70. Team D is at 80 points. Team F is also at 80. Team G is at 100 points. And leading this quiz slightly is Team H with the score of 120 points. Still early days. Yes? Your team D, right? You gave the second answer correct on pounds. Salmonella. And then you gave rinderpest correct. Mink correct. Endosulfan correct. 20, 40, 60, 70. केल्विन शॉबे इनका राइट है तो केल्विन शॉबे नेगेटिव इन्हें नहीं मिलता है फॉर टीम डी हाँ टीम डी है ना हाँ टीम डी का प्लस या राइट राइट या हाँ नहीं नहीं वो टीम ई का है ए और ई का है हाँ ई को नेगेटिव करो सर यहाँ पे पॉजिटिव आई एम सॉरी टीम डी इज एट 90 पॉइंट्स एंड दैट नेगेटिव वाज फॉर टीम ई टीम ई इज एट 70 yes you have a problem too Okay, yes. Team D is at 110 points. Team E is at 40 points. Your negative actually was accidentally given to them. And yes, what is your problem, Team F? Confusogram 10, Salmonella 20, Koala you did not give, Rinderpest 20, Mink 10, Vargiscurin 20, and Schwab 20. 20, 40, 50, 60, 70, 80, 90, 100 points. Sorry, totaling mistake. 100 points, good. 40, 50, 60, 70, 80, 90, 100. 100 points. Confusogram 10.
110 ऐसा कुछ आएगा
Confusogram 10.
Yes sir.
Salmonella 20. Rinderpest 20. Mink 10. Endosulfan you did not give. Curian 20. Calvin 20. That's it. 20, 40, 50, 60, 70, 80, 90, 100 points.
Yes.
Team F 100. Total mistake. Some totaling mistakes here there. Okay, moving on to round 2. Doesn't matter. Every single answer that the participant is given is recorded over there. So even if there might be a totaling mistake, eventually we'll be able to track which team actually gave the answer. So there is no scope of error eventually. So the next round is called Choose Your Topic round. Can you go on to the next slide, please? We have 16 questions here. Teams have to choose the topic on which they want to answer. Rest of the rules remain same. Pounds applies for everybody. Anti-clockwise pattern will be followed here. So the first question will be a direct to team H. So here are your topics, team H. Make a choice. Beverages, ecological dependence, nicknames, AMR, history, paintings, Indian cinema, military history, wildlife, chemistry, vaccinations, global projects, Hollywood, failed campaigns, African terms, and mythology. Make a choice, please.
Nickname.
Okay, so Team H has chosen nicknames. In a quiz on One Health, the burl flower tree, also known as Kadamba, holds a lot of significance. The extracts of this tree bark and leaves have exhibited significant antimicrobial activity and have proved to be effective against bacterial and fungal pathogens, apart from being used for skin infections and wounds in traditional medicine. And its leaves are also used as a common cattle fodder in India. So a lot of One Health topics in the first paragraph itself. The Kadamba flower, despite of being a huge source of nectar for several pollinators, can also be a part of a quiz on One Health for an entirely different reason owing to its recently acquired nickname. What is the new nickname of the Kadamba flower? Anybody wants to pounce? So as you can see, this particular flower has a lot of properties and hence it will qualify in multiple ways in a One Health quiz. But we are talking about a recently acquired nickname according to most of the people, and that has something to do with the image. Direct for team H. Anybody wants to pounce? Nobody is pouncing. You just have to tell me what is this new nickname of Kadamba flower owing to the image. Okay, closing the pounce in 3, 2, and 1. Team H, what is your answer?
Corona flower.
Why?
Because of its shape.
Its shape?
Yes, it
Plus
resembles.
20 to team H. The answer was staring you on your face. It was so similar to coronavirus. That's why it's now called corona flower. Plus 20 to team H. And a direct question to team G. Make a choice, please, team G.
Global projects.
Global projects. Global projects is what team G has chosen. The tricycle protocol refers to a global project by the WHO to monitor one indicator, X, across the human, animal, and environmental sectors. The protocol includes setting standard methodologies across the human food chain and environmental sectors so as to facilitate the establishment of the integrated multi-sectoral surveillance on AMR. The indicator in question, X, refers to a type of bacteria that normally lives in the intestines of humans and animals, with most strains being harmless and beneficial for digestion. X can be grown and cultured easily and inexpensively in a laboratory setting and has been intensively investigated for over 60 years, making it the perfect specimen for this protocol. Identify X. I was sitting there and professor was giving a lecture. I happened to hear this a lot of times. So how many teams are pouncing? 2 teams. Okay. Okay. So team D, okay, so 3 teams have pounced. Team B, Team D, and Team H. B, D, and H have pounced. Any other team wants to pounce? It is a direct for Team G. Closing the pounce in 3, 2, and 1. Yes, Team G, what is your answer?
E. coli.
We'll fetch you a plus 20. Round of applause, please. So B, D, G, and H, they get a plus 20 each. B, D, G, H. Direct to Team F. It's a direct for Team F. Please make a choice. Team F. On the mic, please.
AMR.
AMR is what Team F has chosen. Basically, you have to identify the gentleman over there. I'll read out the question for you. When X was awarded the Nobel Prize in 1945 for his discovery that changed the world, he warned against the improper use of this discovery as it might lead to resistant bacteria. His prediction came true when Staphylococcus aureus was identified, which was probably the first case of antimicrobial resistance. Who was X? You just have to identify the gentleman there, awarded Nobel Prize in 1945 for a discovery that changed the world. Who all are bouncing? Okay. Okay. C and D have pounced so far. Anybody else? Okay. So C, D, G, and H have pounced. Anybody else wants to pounce? Okay, team B. Okay. So B, C, D, H. B, C, G, H. Am I right? B, B, D. Okay. So B, C, D, G, H. They have pounced. Anybody else has? B, C, D, G, H have pounced. And pounce is now over. It is a direct for team F. Yes, team F.
Alexander Fleming.
Alexander Fleming will fetch you a plus 20. So Fleming had predicted AMR before AMR was really cool. So yes, every team that pounced, they get a plus 20 and also a plus 20 for team F. Direct to team E now. Team E, please make a choice.
Vaccination.
Vaccinations is what team E has chosen. Ukrainian-born Dr. Valdemar Mordeshoy, I'm sorry if my pronunciation is incorrect, X's legacy is celebrated throughout the world, particularly in India, thanks to the vaccines developed by him. In 1896, when bubonic plague struck Mumbai, nearly 25,000 people lost their lives in the first year itself. But within a short time, Dr. X managed to create a vaccine that saved the lives of nearly 40 lakh people who got vaccinated. For his contributions towards the development of the vaccines of bubonic plague and cholera, Joseph Lister called him the savior of humanity, and the plague laboratory of Mumbai was named as X Institute after him. Just identify the gentleman. So Mumbai, as we know today, is there due to this bubonic plague epidemic that took place in 1896. A lot of land under sea was taken over, a lot of buildings were destructed, and Bombay Municipal Corporation was formed after bubonic plague. You just have to tell me the name of the person who eventually found out the solution to this problem. Now tell me who all are pouncing. A, B, C, D, anybody bouncing? E, F, G, H, anybody bouncing? H is bouncing. Anybody else? F, okay, F is also bouncing. Okay. So F and H have bounced. It is a direct to team E. Yes, team E, what is your answer? Team E is not trying. Passing on to team D. Team C. No negative, on wrong.
Sir, Jon Snow.
Jon Snow. You know nothing. Team B. Team A. Team H has already pounced. Team G.
Hopkins Institute.
Is there an institute called Hopkins Institute? It is there in Mumbai. Plus 20 to every team that has pounced. That is H gets a plus 20. G gets a plus 20. Yes, and F. F, G, and H, they get a plus 20. None of you pounds, right? Okay, your chance is over. It's now Team D's chance. Make a choice, please, Team D. Plenty of topics. Please be quick because 2 more rounds are there. On the mic, please, so that everybody can hear.
Wildlife.
Okay, so Team D has chosen wildlife. Here is your question, Team D. One main reason for the failure of the Indo-Iran talks about the living being X in the 2000s was its critically endangered status and the fact that it's also seen as a cultural heritage symbol. Referred to in Persian as Yus Palang, X has featured prominently in ancient Persian art forms and literary works, but little has been done to preserve its presence over the years. Apart from illegal hunting and traffic accidents, the US imposed sanctions on Iran happens to be one of the major reasons for X's reducing numbers. In 2014, in order to create international awareness about X, the Iranian football team featured X on its official jerseys, but even in 2025, only 20 X's are reportedly living in the wild, according to the Iran's Department of Environment. What is X? And I would recommend a specific answer. It's a question on wildlife. Anybody wants to pounce? So a cultural symbol of Iran, and there were talks of the Indian government with Iran, which eventually failed. And those talks were about this living being. Despite of severe attempts, only 20 X's are reportedly living in the wild, according to Iran government's own environment website. What are we talking about? Direct to Team D. Anybody pouncing? 3, 2, and 1. Team D. What is your answer? Okay, Team D is passing on to Team C. Would you like to try? Team B? Team A? Team H? Team G? Team F? Team E? Okay. Anybody from the audience? Can you be a bit more specific?
Asiatic cheetah.
A round of applause for Sir, please. It is indeed the Asiatic cheetah. India actually was in talks with Iran to acquire the Asiatic cheetah. It failed. No points to anybody. Direct to Team C. Everybody please answer on the mic, please, so that the audience can also get to hear what you're saying.
Sir, history.
History is what Team C has chosen. In his lifetime, Sir Spencer Harcourt Butler was a polarizing figure with both lovers and haters of his legacy. During his time as the governor of the United Provinces, that is Uttar Pradesh, he is known for his initiatives on industrial advancement and education. However, his role in the partition of Burma from India and his inability to control communal tensions in Burma have often been questioned. In a quiz on One Health, Sir Butler will always be remembered for having established an organization in 1911 and also chairing its first meeting. By what name do we know this organization today? So historians will tell you Sir Harcourt Butler was the governor of Burma. He, when India became a nation, he played a major role in convincing the states, princely states, to make a decision. And he could do nothing about the communal tensions that happened in Burma when he was the governor there. But again, we are not in a history quiz, we are in a One Health quiz. So here we know him for being the one who established an organization in 1911. We know this organization by what name today? That is the question. Okay, anybody pouncing? Team F is pouncing. Okay. So only team F has pounced. I am closing the pounce in 3, 2, and 1. Pounce is now closed. Team C, what is your answer?
Sir, ICMR.
ICMR. And by what name did we know it then?
Sir, Indian Research Fund Association, IRFA.
Plus 20 to team C and team F. For answering it correctly, it was indeed ICMR. The answer was there in the question below the logos. So team C and team F, they get a plus 20 each. Direct question to team B. Team B. On the mic, please.
So military history.
Military history is what team B has chosen. One of the major producers of sugar and coffee, Haiti was always an important colony for the Europeans, which fell from the hands of the French in the year 1791. In order to recapture it, Napoleon Bonaparte sent an army of close to 60,000 soldiers under the famed general Charles Leclerc, who was also Bonaparte's brother-in-law. But within a year, most of the army, including Leclerc, died due to an epidemic, which resulted in French defeat. With Haiti's declaration of independence, Napoleon also ended his ambitions in America. Which epidemic are we talking about that indirectly liberated a nation? So Napoleon sent his brother-in-law to recapture Haiti, and he failed. Not because of the soldiers of Haiti, but because of this disease.
Sir, is there negative marking if we answer wrong?
On the direct, there is never a negative marking except for the buzzer round.
Okay.
But if you're pouncing, then make sure you know the answer. It's your direct. Team B, please wait before you answer. Anybody wants to pounce? Okay. Team D is pouncing. Okay. So team D has pounced. Anybody else wants to pounce? Closing the pounce in 3, 2, and 1.
So plague.
It is not plague. That's what they two answered. Team A. Team H.
Scrub typhus.
Not cholera. Team F. Team G.
Kalazar.
Kalazar. Team D has already pounced. Only team C is left. Yes.
Spanish flu.
No. Anybody from the audience would like to try? It is indeed yellow fever. Big round of applause for sir. And unfortunately, a negative 20 to team D. Team D gets a negative 20. No other team gets any points. So team A, it's your direct now. बाकी टीम से सोच के रख लो कौन सा लेना है पहले. Yes, team A.
Mythology.
Team A has chosen mythology. Hadkai Mata is a folk deity worshipped in parts of Gujarat and is believed to be the protector of humans as well as another living species, which is not just her Vahan, but also drives her chariot. According to legends, Hadkai Mata punishes sinners by giving them a particular disease, and if the sinner does not immediately visit her shrine and repent his actions, the disease eventually takes the life of the victim. Although it cannot be said if the legend of Hadkai Mata has played a role in reduction of the sins committed, it certainly can be said that the disease which kills more than 10,000 people every year in India is often not taken seriously by not providing timely medical attention to the patients or sinners. What disease are we talking about? Wait. Anybody wants to bounce? So this is a very serious issue. And there are hardcore devotees who get this particular disease and they don't see a doctor. They go to a shrine, which is not something that needs to be encouraged. So anybody wants to pounce? It kills 10,000 people every year, approximately, nearly or more. Direct to team A. Anybody wants to pounce? Okay, team F is pouncing. Okay. So team F has pounced. It's a direct for team A. Anybody else pouncing? 3, 2.
Smallpox.
Chicken pox. Okay, and the living being chicken. Okay, team H. Team H is not trying. Team G. Tuberculosis, no. Team F has already pounced. No. It's not smallpox. Team D. Team A has attempted. Anybody else? Nobody paid heed to the first line. Believed to be the protector of humans, which is not just her Vahan, but also drives her chariot. Now, anybody in the audience? It is indeed rabies. Hadkai Mata has her Vahan as a dog, and it is often believed by the devotees that if you mistreat an animal or if you commit a sin, आपको rabies हो जाएगा. And just go to the temple, try a thread, and it will be all right, which is not true. You must see a doctor if such a thing happens. Unfortunately, a negative 20 to team F. Nobody gets any points. So team H, it's once again your direct.
Ecological dependence.
Ecological dependence is what team H has chosen. In the 1970s, a theory was proposed that the Tambalacock trees of this nation on the southeastern coast of Africa were all close to 300 years old and might be extinct soon. And the reason for this extinction of this tree, according to this theory, was the extinction of another living being, X, which once ate the seeds of this tree, and after passing through its digestive tract, the same seeds were able to germinate. The popularity of this theory resulted in the tree also being called the X tree. However, scientists have now proven that this tree seeds can also be germinated when passed through the digestive tracts of several animals. And although the tree is still not extinct, it continues to be listed by IUCN as critically endangered. What is X? So it was believed that this tree will eventually become extinct because the only way its seeds will germinate is by passing through the digestive tract of some other living being which was extinct. Later, this theory proved to be false. The seed is capable of germination by going through the digestive tracts of some other animals as well. So the theory of the 1970s was so popular that this tree even today is called X tree, although its official name is Tambalacock tree. Just tell me the living being that got extinct. Anybody wants to pounce? How many animals or birds or any living species do you know that got extinct? Those are from the southeastern coast of Africa. I'm closing the pounce in 5, 4, 3, 2, and 1. Team H, what is your answer?
Dodo bird.
As dead as a dodo. Will fetch you a plus 20. Tambalacock trees are also called as dodo trees because the association you already know. No teams gets any points except for team H. Team G, it's your direct now. On the mic, if you can make the choice.
Failed campaigns.
Failed campaigns is what team G chooses. The Great Blank War of 1932 was a military operation initiated by the Australian government to counter the terrorist designs of the enemy in quotes, who were supposedly 20,000 in number. The Royal Australian Artillery soldiers equipped with Lewis guns participated in this war that lasted for over a month. Despite of killing over 900 of their opponents and not suffering a single casualty, the Australian army still lost the war and also its reputation, along with facing global criticism. The commander of the Royal Australian Artillery's 7th Heavy Artillery Division, Major Murdith, later praised their opponents by comparing them with the Zulus and stated that if the Australian army had their skills, they would have been the most invulnerable military force in the world. Who were the opponents of the Australian army in this particular war in quotes? The Great Blank War of 1932 was a failed campaign initiated by the Australian army. Despite of killing 900 opponents and not having suffered a single casualty, the army of Australia ended up losing the war and also faced global criticism and also loss of respect. And the commander kept on praising their opponents. Like if the Australian army had this particular skill, they would have been the strongest military force in the world. Anybody wants to bounce? Team H is bouncing. Okay, one team has bounced. It's your direct right. Any other team wants to bounce? 3, 2, 1. Go ahead, team G.
Chinese.
Chinese. Okay. The Great Chinese War. Why will that be a question in a One Health quiz?
Britain.
Britain again.
Iran.
You all are thinking in one direction. Team D.
Pass.
Team D is passing. Team C. Team B. Team A. Team H has already pounced. Anybody from the audience would like to try? The Great Emu War. Round of applause for sirs who answered it correctly. And also to Team H. They answered it correctly on pounce. The army, despite of its might, was not able to exterminate the emus. So the Austrian army eventually surrendered. Direct to Team F now. Yes, Team F, make a choice, please. Indian cinema is what Team F has chosen. Again, people will be wondering, what is the question on Indian cinema doing in a One Health quiz? Let's see. There is Hollywood too. So Virus is a 2019 Malayalam movie based on a real-life virus outbreak in Kerala in 2018. The movie makers told the story of how the government, doctors, and the citizens played a major role in controlling the major outbreak, despite of having no prior knowledge about it, and thus averting a major tragedy. The movie was unanimously hailed by the critics, and its smashing success at the box office also educated the common people of the state about concepts like quarantine, social distancing, and about the overall functioning of a virus, all of which proved to be important knowledge in the COVID pandemic later. Which virus outbreak was the subject of the movie? A lot of things actually happened in Kerala. One of them was the 2018 virus outbreak that eventually inspired this particular movie called Virus. So some teams are pouncing. Okay. Okay, so G and H have pounced direct for Team F. Anybody else pouncing? Okay. 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 So C, D, G and H. C, D, G and H have pounced. Other teams, do you want to pounce? Closing the pounce in 3, 2, and 1. Team F, what is your answer? Nipah virus will fetch you a plus 20. Big round of applause for all the teams, please. C, D, F, G, H. C, D, F, G, H. मैं भूल गया A आता है कि C आता है बाद में. So C, D, F, G, and H get a plus 20 each. It's a direct for Team E now. Make a choice, Team E. Beverages, chemistry, African terms, paintings, Hollywood. And chemistry. Okay. Chemistry is what Team E has chosen. Although this product was created by Austrian chemist Ottmar Ziedler in 1874. Its properties were a Nobel Prize winning discovery of the late 1930s by Swiss chemist Paul Hermann Müller. Initially, when the WHO recommended the usage of this entity against the spread of malaria, the resounding success that followed was hailed unanimously, but its effects on environment were severe, especially on predatory birds and fishes. And when the effects on humans were also evident, people started questioning its usage. In many parts of the world, the Anopheles mosquitoes developed resistance to this entity and became doubly dangerous. Although banned in many parts of the world, this entity continues to be used in a very restrictive manner. What entity are we talking about? Any chemistry student would be able to tell you the answer, even if you're not from chemistry background. There are several things that I've mentioned here. Its impact on malaria and on human beings. It is still there, but its usage is very restricted. A Nobel Prize winning discovery. Direct to team E. Anybody wants to pounce? I'm sure all of you know of this. It's not something new. Team C is pouncing. Team A is also pouncing. Okay. 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 Pouncing. Okay. B is also pouncing. Okay, so A has pounced, B has pounced, C has pounced. You haven't. D hasn't pounced. आपका direct है. You have pounced. दिखाइए. Okay. Okay. So every team has pounced except for team D, right? Yes, team E. What is your answer?
Quinonon.
Quinonon is not correct.
Hydroquinonon.
No, team D.
DDT.
DDT. And what is its full form? Will fetch you a plus 20. The answer is indeed dichloro, diphenyl, trichloroethane. Points pattern is a bit confusing. I'll tell you, team A gets a plus 20. Team B gets a plus 20. Team C, unfortunately, gets a negative 20. Plus 20 to team D for answering directly on bounce. Team E gets no points. Team F gets a negative 20. Plus 20 to team G and a negative 20, probably their first negative 20 for team H. DDT. So team D, it's your direct now. Make a choice, please. Beverages. Beverages. A very easy beverage awaits you there. While the name of this beer brand being same as an unwanted health occurrence was a coincidence, which was largely joked upon by the internet users, situation suddenly became serious when the sales of the beer dipped as people supposedly began to link both the names with each other. The beer brand eventually had to issue a statement that their product's usage did not result in the issue being talked about, and even the Delhi High Court restrained companies creating ads linking this beer to the issue. What is the name of this beer that owes its origin to the Spanish word for a particular ornament? So we are talking about a beer brand and an unwanted health occurrence and the name of the beer brand happen to be same. So the internet users eventually created memes out of it, but situation got out of hand when people actually started to associate this beer brand with the unwanted health issue. and the sales started dipping down. So this beer brand company had to issue a statement that our brand has nothing to do with what you're talking about. What is the name of the beer brand? That is the question. Okay, one team is bouncing. Everybody understood the question, right? Okay, team H has bounced. Direct to team D. Anybody else wants to bounce? You'll curse yourself after seeing the answer. Nobody is bouncing. 3, 2, 1, go ahead, team D. I'm sure you know.
Budweiser.
Budweiser. But why will that be an issue? Team C, you want to try? Okay. Team B.
Corona.
There is a brand called Corona. There is plus 20 to team B and also team H. Corona beer was the brand, and the sales dipped because people actually thought while drinking Corona beer, you might actually get Corona. So that's the point system. Team H gets a plus 20 and team B gets a plus 20. It's a direct for team C now. Please make a choice.
Paintings.
Team C has chosen paintings. A very long but interesting question awaits you, Team C. This painting by Thomas Hickey from 1805 shows the two wives and the grandmother of King Krishnaraja Vodayar III of Mysore modeling for X, and is the only one of its kind to exist. Invented in 1798 and thus still a fairly new product, X was met with suspicion and often fierce resistance in India, since it was seen as a tool by which the British were contaminating Indians and disrupting their religious beliefs. In this hostile scenario, the British turned to the Vodayars, whom they had reinstated to the throne after defeating Tipu Sultan. After months of convincing, the Vodayars finally accepted the British idea of a portrait to promote X, with the three royal women as models, ceding that the humiliation of using their house as an advertising campaign was far outweighed by the overall good it would do. The portrait itself shows the three women dressed in over-the-top royal gear and flaunting X, with minimal loss of dignity. Identify X, the product which the British were trying to desperately promote. So I'll shorten this question for you. This painting was made in 1805. The British wanted to promote something in India. But what happened was the Indian religious beliefs, they were pretty skeptical about this idea. They thought the British are doing something crazy so that our religion gets compromised. So the British could not get hold of people for this. So they managed to convince this Vodayar dynasty of Mysore to allow the women of their house to pose as models for this particular product. It's not really a product. You just tell me a generic idea. What are we talking about? So actually, even the kings were not really convinced for sending out the women from their folk to campaign for this, but they were sure that the outcome would eventually be good. Despite of loss of dignity, the outcome will eventually be beneficial to humanity. That the kings were convinced. That's why they went ahead with the idea. We have the painting with us in the next slide. This is the painting. Everybody understood the question, right? The British wanted to promote something. And this royal woman, they managed to model for that idea or that product. There are certain clues in the image. What are we talking about? Can we go back to the question, please? Some important timelines mentioned over there, 1798, 1805. Okay, so nobody is pouncing, it seems. Pounce closing in 3, 2, and 1. It's a direct for team C, right? Okay, can we go back to the image? As I said, there are certain clues in the image. Okay, so pounce is now closed. Please go back to the question again. Yes, Team C. You have an answer? Team B? On the mic, please.
Is it Da Vinci?
Da Vinci, 1798. Team A? Team H? Team G? Image, please.
The clothing?
Okay, but again, it's a One Health quiz, right?
Yeah.
Team F? Team E? Team D?
Mysore silk.
Mysore silk, One Health quiz. Anybody from the audience? Smallpox vaccine. And can you tell me something more about what's there in the image?
So she's showing the vaccination patch which you get when you do the vaccination. And I think these are very young generation. So you can see the patch on the people who has taken the vaccine.
Round of applause for ma'am, please. Smallpox vaccination. Nobody got it right. Last but one question of this round is a direct to Team B. African terms or Hollywood?
Hollywood.
Hollywood is what Team B has chosen. Steven Soderbergh's thriller movie X, released in 2011, is believed to be the most realistically made movies on a pandemic. Inspired by the swine flu outbreak of 2009, this movie very systematically explains the working of a pandemic and highlights the concept of zoonotic spillover. X features several A-list actors like Kate Winslet, Matt Damon, Jude Law, among other Oscar-winning actors, and was a grand success at the time of its release, and also attracted renewed interest during the COVID-19 pandemic. What is X? A name that originates partially from Latin, meaning to touch. We have a video. This is basically the spoiler, but still go ahead and watch it. Now, can we move on to the question? So as you understand, what you saw in the video happens to be what zoonotic spillover basically is. But you have to tell me the name of the movie, and the only clue there is the last line. It originates partially from Latin, meaning to touch. It's a direct to Team B. Anybody wants to pounce? Closing pounce. 3, 2, 1. Team B, go ahead.
So we don't know.
Okay. Team A.
The pandemic.
Okay. Team H. None of you have seen the movie. Okay. Team G, Team F, Team E, and Team D. Team C. Anybody from the audience? Contagion is indeed right. A big round of applause for the audience, please. No points to anybody. Last question of this round is a direct to Team A. So Team A, what do you choose? African terms. I see you're an expert in African terminologies. Okay. Although this viral disease is believed to have existed since 1779, it was recorded for the first time only in 1955. The name of this disease originates from Makonde language, roughly meaning that which bends. referring to the out of shape posture of people owing to joint pain, one of its important symptoms. However, the name has often been incorrectly attributed to the Swahili language. Identify this viral disease which created a lot of panic in India in the mid 2000s. Derek for team A. We all are aware of this particular disease. We also know it's something to do with Africa. We might not be knowing the Makonde language part or its meaning, that which bends. Okay, now tell me who all are bouncing. Derek for A, B, C, D, do you want to bounce? An African term. How will it be this? Okay, so team F has bounced. Team H has also bounced. Anybody else wants to bounce? G is also bouncing. Okay, F, G, and H, they have bounced. Closing the bounce in 3, 2, and 1. Go ahead, team A. Give me any African sounding name.
Chikungunya.
Chikungunya. Is it African sounding? It is. Plus 20 to team A, team H, team G, and unfortunately a negative 20 to team F. Team F gets a negative 20, A gets a plus 20, G and H, they get a plus 20 each. So that is the end of this round. 2 more rounds to go. So I hope you guys are finding this quiz interesting. You can be a part of Nagpur Quiz Club where you can participate in many more topics with questions similar to this pattern. These are not you know it or you don't know it type of questions. We try to make our questions paragraph based so that there is more than one way to reach out to the answer. Again, we are manually counting the scores. In case your tally differs from us, please tell us. We have all the answers recorded. And if there is any discrepancy, we can take care of it immediately. So the total score of team A is 60 points. You scored 40 in this round and 20 earlier. Round of applause for team A, please. Team B, you scored 110 points earlier and 80 in this round, taking your total score to 190. Team C, you scored 60 earlier and 40 in this round, total score 100. Team D scored 110 points earlier and 60 in this round, 170 total. Team E scored 40 points earlier, total score 40. Team F scored 100 earlier and 20 in this round, total score 120. Team G scored 100 earlier and 120 in this round. Total score 220. Team H scored 120 earlier and 160 in this round. Total score 280. Okay, so this round is the buzzer round. The questions will not be there on the PPT. The quizmaster will be asking the questions verbally. You will have to listen to me very carefully. And the first team that attempts the buzzer, I'll get to see the number over there. Only that team will be allowed to attempt. You'll either score plus 20 for correct. And after pressing the buzzer, if you don't give me an answer or give me an incorrect answer, you will get a negative 20. It's either plus 20 or negative 20 if you press the buzzer, nothing else. Am I clear? And the questions won't be there on the screen. I'll be reading them out to you. There are 10 questions overall. Just check one by one if the buzzer is working properly. Team A, please press your buzzer. Okay. Team B, okay. Team C. Team D. Team E. Team F. Team G. Team H. Okay, another thing, answer on the mic only when I ask you to answer. There shouldn't be a confusion, like if team H has actually pressed the buzzer and team G is the one answering, then it'll be trouble. No, it's not possible, and neither can you pounce. You'll have to listen to me and press the buzzer. But again, if you don't let me ask the question completely, if you press the buzzer in between, I'll stop asking that question. You'll have to answer right away. Okay. You can, if you are sure of the answer, you can go ahead, but I'll not ask when I hear the buzzer. Question number 1, which organization was originally called as Indian Research Fund Association? Team C, what is your answer?
The ICMR.
Team C gets a plus 20. We had already discussed it in the previous round. Question number 2. In India, the first center for One Health was established in, yes?
NIOH Nagpur.
Plus 20. That was my question. Yeah, but had you pressed first, your number would have appeared. Press it now. It's working fine. Okay. Reset kar diya. Question number 3. In order to raise global awareness about the interconnectedness of human, animal, and environmental health, on which day is the International One Health Day? Yes, Team C?
November 3.
Plus 20 again to Team C. Question number 4. Every team needs to be alert. How do we better know the phenomenon where microorganisms evolve to resist the effect? Team C.
Microbial resistance.
Plus 20 to Team C. So what Team C is doing is they are listening to keywords and applying their brains. Every team is requested to do that. Okay. This question I'm sure Team C will associate even more. In the 4 C's of One Health. Yes, Team 4. You have to give me one answer and I will not continue the question.
Coordination.
I'll now ask the question for no points. In the 4 C's of One Health refers to the essential components that transform the theoretical concept of One Health into practical coordinated action across human, animal, and environmental sectors to achieve optimal health outcomes for all. What is the first C in this concept? If the other C's are coordination, collaboration, and capacity building, no points now. Unfortunately, Team C gets a negative 20. What I intended to ask Team D, Team D. Team D, Team D. So Team D jumped the gun. The answer indeed was communication. Question number 6. Which German physician is considered the father of One Health concept? Yes, team. Father of One Health concept, who famously stated, between animal and human medicine, there is no dividing line. The answer is Rudolf Virchow. A negative 20 to Team F. Question number 7. Which European city will be the host of the upcoming International One Health Conference? Sorry?
Portugal.
European city was what I asked. The answer is Copenhagen. Negative 20 to Team G. Question number 8. COHEART, Center for One Health Education, Advocacy, Research and Training, is an autonomous center working under the aegis of blank veterinary and yes, Kerala was indeed what I was looking for. A plus 20 to Team G. Question number 9. What is the term that scientists use as a placeholder for any unknown infectious agent which would cause a future outbreak, epidemic, or even a pandemic? Yes. Pathogen X will fetch you a plus 20. Plus 20 to Team G. Last question of this round. Which disease is also known as hydrophobia? C. Will fetch you a plus 20. अभी दबा के कोई फायदा नहीं. So in this round, Team A and B did not score any points. So your score remains the same as they were earlier. Team C managed to score 100 points, doubling their score to 200 in this round. A big round of applause for Team C, please. Team D got 1 negative, taking their total score to 150. Team E did not score anything. Team F also secured a negative 20, taking their score to 100. Team G got a plus 20 and a minus 20, taking their total score to 240 points. 220 and 1, right. 20 points overall. You scored 2 right and 1 incorrect. So 20 points added on to your score. And you're now at 240. Team H also scored no points. 280. Moving on to the final 8, it's not 10, it's 8 questions of this round. It's called Final Frontier. 8 questions. Here, there are 3 types of pouncing that is allowed. Free hit pounce, that is plus 10 or 0. Normal pounce, that we saw earlier, plus 20, minus 20. And there is something called power play pounce, plus 30, minus 30. While pouncing, please ensure what type of pounce you're doing. You can score as many as 30, but you can also lose minus 30, and you can have an option of free hit as well, where you can pounce and still not get a negative despite of giving me a wrong answer. For the direct, the point scheme is plus 20 as it was earlier. Understood? Okay, so the first question is a direct for team A. Here is your direct question, team A. The ex-Congo hemorrhagic fever is a viral disease, the symptoms of which include fever, muscle pains, headache, vomiting, diarrhea, and bleeding into the skin. This virus was initially detected in the 40s when Soviet troops and local civilians in X experienced a severe hemorrhagic illness. Decades later, during the 1960s, a similar virus was identified in the Democratic Republic of Congo, and the two viruses were found to share antigenic properties, leading to their classification under the unified ex-Congo hemorrhagic fever. Identify the other place named in the disease, X, that has been in the news since a decade due to geopolitical tensions. So we are looking forward to a place. X is a place, and ex-Congo hemorrhagic fever is the disease. Derek for team A, how many of you want to pounce? Please tell me whether it's a normal pounce or a free hit or a power play. No, you can't. After direct, you will get 20 irrespective. Power play, okay. Doesn't matter. Okay. Power play, normal, free hit. Power play. Okay, go for it. Okay. Power play. Okay. Team E, you're bouncing? Free hit. Okay. Okay. Power play, free hit. Okay. Power play, free hit. Power play. Dikhao. Okay. Power play. Okay. So everybody has pounced. Only team E has hit a free hit. Everybody else has answered correctly. Go ahead, team A.
Crimean
Congo will fetch team A plus 20. Every other team gets a plus 30 except for team E, which gets a plus 10. Team E, you should have power played. A gets a 20. Gets 30 except for team E, which gets a plus 10. Team B, it's your direct now. The Center for One Health in India implements 4 national health programs, which are for the following diseases: rabies, zoonosis, snakebite, and X. X is a bacterial disease prevalent in tropical and subtropical areas like Kerala, Tamil Nadu, Gujarat, Karnataka, and Maharashtra, particularly during monsoon seasons and post-floods. Transmission of X occurs through exposure to urine from infected animals, such as rodents, dogs, and cattle, often via contaminated water and soil. Identify the disease. Derek for team B, anybody wants to pounce? Team A is not pouncing. Team C, power play. Yeah, free hit. Okay. Power play or free hit? Okay. Power play. Okay. Good decision. Okay. 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 So every team has pounced except for team A. Team A, do you want to pounce? I don't want to bully you again. If you're sure you can pounce, I'll give you 3 seconds to make up your mind. 3, 2, and 1. Sorry, time up. Go ahead with your answer, team B.
Leptospirosis.
Plus 20 to team B and a plus 30 to everybody else except for team A. Team C, it's your direct now. Again, a video question. The X birds were used in China for fishing purposes since the 6th century. After partially releasing these kill birds underwater, within no time they would get hold of a large fish, which the birds would try to swallow. But since a loose ring is already tied on their necks, they are unable to swallow it. The fishes are then retrieved from their throats by the fishermen who eventually sell these fishes. Although highly successful in its time as a source of revenue, the X fishing is now done primarily to entertain tourists. Which birds are we talking about? We have a video. This is a BBC documentary called Wild China. This is the bird. So something is tied to the neck of that particular bird so that it does not get to swallow the fish. So the bird tries to swallow the fish after getting hold of it, but it can't due to the knot tied on its neck. So eventually the fisherman gets to capture the fish. As I said earlier, this used to be a source of livelihood for many Chinese, Japanese, and Korean fishers, also in Thailand. But today nobody really follows it. This is done basically to show the tourists. You just have to tell me what birds are these. Direct for team C. Anybody wants to pounce? Can we go back to the question, Anushka? Just identify the bird species. Even a generic answer is acceptable. But don't say something like a blackbird. That is not a generic answer. Direct for team C. Anybody wants to pounce? It's okay. You're free hit. Okay. It will come to you if they don't answer. Free hit. Team B opts for a free hit. Okay. You want to free hit? Okay. So two teams have used the free hit option. Closing the pounce in 3. Free hit, yes. Free hit. Okay. So team G has also used a free hit. Free hit. Okay. So who all have pounced? Team B, Team D, GH. B, D, GH have pounced and none of them got it right. But no negative since you did not use a power play or a normal pounce. It is a direct to Team C. Go ahead with your answer. Team C. Team C is not trying. It will not pass to Team D, Team E, Team F. Team G has already pounced. H has already pounced. Team A. Team B has already pounced. Anybody from the audience? Yes, it is indeed cormorant, greater cormorant, lesser cormorant, any cormorant. Nobody gets it right on pounce, but no negatives to anybody. Question number 4 is a direct to team D. Nobody gets any points. Next question is a direct to team D. The first case of this hemorrhagic fever was discovered in the forest area near Kattinakare village in Karnataka, 1957. You are from Karnataka, right? It is unfortunate that you got it as a direct. While it was believed to be an epizootic disease among monkeys, soon the same virus was found to be present in humans And today, more than 400 people are affected every year by this disease. Although commonly referred to as monkey fever, what is the name of this zoonotic disease? I hope it is a zoonotic disease. Okay, everybody is bouncing. Power play or free hit? Normal. Okay. Power play. What if I tell you I want the correct spelling? I'm just kidding. Go ahead. Okay, it is not. Doesn't matter. Okay. Team E, you're not bouncing? Bouncing. You're power play or free hit? Hmm? Power play, 30 minus 30. Normal, 20 minus 20. Free hit, 10-0. Normal. ठीक. It was a free hit. Normal. Okay. Power play. ठीक. Full form लिखिए. I'm okay with. क्यों कर रहे हो? Okay. Okay, so every team has pounced. Okay, so only team E has done a normal pounce. Every other team has power play, and every team gets a plus 30, except for team D, they get a plus 20. Go ahead, team D.
Casino or forest disease.
Yes. Excellent answer by everybody. Okay, I'll clear myself once again. Team D and team E, they get a plus 20 each. D and E get a plus 20 each. D for Delhi, E for elephant. 20 each. Every other team gets a plus 30. E, it's your direct now. One major threat to the very existence of this family of critically endangered animals is diseases transmitted to them through humans. A single human with a cold has the potential to eradicate them entirely if not dealt with properly. To fight this issue, an international collaboration called GAMU or GAHMU played a major role in understanding the impact of the diseases and to prevent them from happening. Some of the national parks that came under their purview are in African nations like Tanzania, Republic of Congo, Cameroon, etc. Which family of animals are we talking about? So I'll tell you, HMU here stands for Health Monitoring Unit. You have to tell me what is GA. Anybody wants to pounce? These animals are found in Africa, Tanzania, Republic of Congo, Cameroon, etc. Critically endangered animals. Direct to team E. Team F is bouncing. Power play or free hit or normal. Okay. Okay. Normal or free hit. Free hit. Okay. Free hit or normal or power play. Free hit. Free hit is plus 20. Why will that be GA? Okay. So team B has pounced, team H has pounced, and team G has pounced. A, G, H have pounced. It happens to be the direct for team E. Yes, team E. What is your answer? GA. Okay. Team F. Team G has pounced. Team H has pounced. Team A.
Gorilla.
Gorilla will fetch you a plus 20. The answer is actually great apes. But again, gorilla falls in that category of great apes. Excellent answer by team A. They get a plus 20. And also team H, they too get a plus Free hit. They get a plus 10. Normal, no, they get normal, 20, 20. 20 for team A and team H. Team H actually gave me great apes as the answer. Big round of applause for both the teams, please. Team F, it's your direct now. No negative, is it? Although believed by many to be named after a river, the X virus was named after a district in Uganda in 1937 and happens to be the most commonly reported disease spread by mosquitoes in America. Apart from humans, the X can also be lethal to horses, and in 2002, as many as 15,257 horses were infected with X in a major outbreak. What is X? It is direct for team F. There is a clue in the first line, believed by many to be named after a river, but in reality it is named after a district. Or play normal, free hit. Normal. Okay. और play करना चाहिए था. Anybody else bouncing? Or play normal. अभी बहुत देर हो चुकी. Or play, go ahead. Okay. So 2 teams have pounced so far. Free hit. आपका direct है. Free hit, power play, normal. Normal. Free hit, power play, normal. Okay. अच्छा हुआ free hit लिया. Yeah. So yes, only team D left. Power play, normal, free hit. Normal. Okay. Every team has pounced, right? Anybody here? Okay, except for team A, every other team has pounced. This is the direct for team F. Yes, team F. What is your answer?
Sorry, Ebola virus. Ebola.
Ebola. Believed by many to be named after, I don't know the existence of an Ebola river. Passing on to team A. Okay, team A is not trying. Every other team has attempted, right? Anybody from the audience? West Nile is indeed correct. Now let's go on to the points pattern. Team A gets nothing. Team B had power play. They get a plus 30. Team C gets a normal. Team C gets a 20. No points. They use a free hit, right? Free hit for team D, 0 points to team D. And you used a normal bounce, right? Or free hit. Okay. Team E gets a plus 20. Team F gets no points. Okay, normal or free hit, normal. Okay, normal, 20 for team G and no points to team H. Next question is the penultimate question of this quiz, direct to team G. The small village near Nagpur, Maharashtra, though largely unknown, gave its name to a virus in 1965 from the blood samples of 2 children who were from this village. This arbovirus primarily spreads by sandflies and mosquitoes and is known for largely targeting children, 329 of whom got infected in an outbreak in 2003, out of which 183 died in parts of Andhra Pradesh and Maharashtra. Its largest outbreak occurred in July 2024 in Gujarat, again affecting children. What is the name of this virus? It's named after a small village in Vidarbha. Okay, कौन सा power play. So team A, power play. Power play for team B. Power play normal. Yeah, free hit. Normal for team C. Power play for team D. Power play normal, free hit. Free hit. It's a free hit for team E. Team F, you want to bounce? Bounce कर रहे हो? Normal. हाँ, कहाँ आंसर आपका? अक्षर इतने खतरनाक है. I'm really worried about your teachers. Okay. क्या था ये? Normal. Okay. -20 इनको. आपका डायरेक्ट है. Power play. Okay. Power play for team H. So every team has pounced. कोई है जिसने नहीं किया? Everybody has. Go ahead with your answer. Team G.
Chandipura virus.
Chandipura virus is indeed correct. Point pattern. Power play for team A, they get a +30. Power play for team B, they also get a +30. Team C used a normal option, they get a +20. Team D, you gave me a wrong answer, right? No, इनका भी correct है, but a normal bounce. Power play for team D, they also get a +30. Free hit and an incorrect answer for team, which team are you? Team E. Team F, normal and a negative for team F, +20 to team G, and a +30 to team H. And finally, the last question of this quiz is a direct to team H. This 25-hectare tropical forest in Entebbe, Uganda, is known for its rich biodiversity. A property of the government-owned research institute, it happens to be entirely protected for ecological study, apart from restricted access to bird watchers. The name of this forest means dense or overgrown in Luganda language, but the name became notorious throughout the world since 2007. What is the name of this forest? So everybody sitting in this room is aware of the name of this forest by something that is named after that forest. We're talking about a term in Luganda language, and this forest is in Entebbe, Uganda. Its meaning being dense or overgrown. Anybody wants to pounce? Derek for team H. Closing the bounce in 3, 2, free hit, okay. Okay. Team E has pounced. Team G is also bouncing. Free hit or free hit, okay. So both the teams have used free hit. Team G and team E. Anybody else? 3, 2, and 1. Yes, team H. No points for you. No points, 0.
Amazon.
Okay. Team A not trying. Team B. You can still guess, right? Team E has already pounced. Not trying. बोल दो कुछ. You have already pounced. Team F has already attempted. Anybody from the audience who's not having a mobile phone in his hand? Yes. Zika is indeed the name of the forest. No points to anybody, but that is the end of this quiz. Till the scorer completes the scoring, I'd like to thank everybody for being very patient in this long quiz. I'm very thankful to Sandeep Chaudhary sir for inviting us again and again here. Thank you, sir. Thank you for inviting us. I hope you invite us again. I'm really sorry for the mistakes that we did today. I'm not somebody from scientific background. Everything I learned from the internet and from some kind people. One of them being Dr. Gayatri Jairaj. She is not here today. This quiz could not have been possible without her support. Thank you, Gayatri. And also my partner, Anirudh Morarka. He's also not here today. Every quiz happens because Anirudh is always there. And a special thanks to Anushka, a leading quizmaster from Nagpur. Thank you so much, my dear friends. So there is a tie for the third position between Team B and Team C. So once again, we'll have to go back to the buzzers. I'll ask a question. Whichever team answers on the buzzer, they win the tiebreaker. But if you give me an incorrect answer or don't answer after pressing the buzzer, the other team automatically wins. Am I clear? Here is your question. Team B and Team C. The buzzers are working, right? Team B buzzer दबाइए एक बार. Okay. Team C. Okay. A common reason for many arbovirus diseases, the genus X was discovered by German entomologist Johann Wilhelm Megen in 1818 and was named after the Greek word for unpleasant. Identify SX, which contributes to nearly 300 deaths in India almost every year. We are talking about a mosquito. A mosquito, which means unpleasant in Greek. Anybody trying? Tiebreaker. Nobody is trying? Okay, the answer is Aedes mosquitoes. Okay, this is an easy one. I want both of you to be alert. In the late 1990s and early 2000s, India experienced a crisis of sort when more than 90% of this living species died. One major reason for this being a cattle painkiller called diclofenac. Team B. Vultures is indeed correct. And Team B manages to secure the third position with the help of tiebreakers. Team B. So finishing 8th today with the score of 120 is Team E from Jaipur. Your score is 120 points. Round of applause for Team E, please. 7th is Team A with the score of 160 points. Round of applause for Team A, please. Finishing 6th with the score of 170 is Team F. And with the score of 260, finishing at 5th position, is Team D. And the 4th position, thanks to tiebreakers, goes to Team C. You two scored 330 points. And finishing second runners up today is Team B with the score of 330 again, with one tiebreaker question. And which team finishes second? Is it Team G or Team H? You're right. Team G finishes second with the score of 370 points. And winning today with the score of 430 points is Team H. Once again, I'd like to highlight quizzing is not about winning or losing. Everybody sitting here knows a little bit extra than what you knew when you entered the room. So keep participating in quizzes, keep conducting more quizzes, and I'm sure your knowledge will rise. Thank you. Thanks once again.
Thank you, sir. I would like to call upon Raja and Kode sir to felicitate Quizmaster. Thank you, sir. Teams, please stay, come off the stage. We will proceed for valedictory. Please proceed, members. Uh,
I would like to call upon dignitaries to stage for validatory function. Uh, uh, Dr. Vinitin Patil, sir, Honorable Vice Chancellor. Dr. Shirish Upadhyay, sir, Dean and Director Instruction, MAFSU. Dr. Kurkure, sir, Director Research, MAFSU. Dr. S.W. Bonde, sir, Director of Extension Education, MAFSU. Dr. Jayant Korde, sir, Associate Dean, NVC. Dr. Barbudde, sir, Director ICR, NMRI, Hyderabad.
As we reach the final chapter of today's One Health Youth Conclave, we commence the valedictory ceremony. We had outstanding participation from students representing 12 universities, and the level of creativity, scientific understanding, and enthusiasm was truly exceptional. Today has been a vibrant amalgamation of science, creativity, learning, and youth leadership. We take a moment to thank all participants, dignitaries, judges, volunteers, and the organizing committee for making this event meaningful and impactful. We'll, we will now announce the first, second, and third prize winners for all the competitions held today. Dignitaries on the dais are requested to kindly present the certificates and awards.
To start with, we'll be announcing the result for One Health Quiz competition. The first prize go to winning team, MAFSU. A big round of applause for all brilliant quiz teams. Second prize goes to PV and Daru, Hyderabad. Third prize goes to team Rajuas, Bikaner. Next is for the essay competition. Here are the winners. First prize goes to Tippa Padmaja, MAFSU, Nagpur. Second prize goes to Harsha Rati, Rajuas, Jaipur. Third prize goes to Saubhagya Kumar Shahu, out, Bhubaneswar. Saubhagya Kumar Shahu. From OUT, Bhubaneswar. Next is e-poster competition. First prize goes to Ganesh A. Kafsu Bidar. Second prize goes to Anurag Kamle, Mafsu, Nagpur. On third is अमोल चौगुले, माफसू, नागपूर. Next we have videography competition. The winning entries are: first is Kondala Apple Lawrence, Piventaru, Hyderabad. Please, anyone from the team come and receive the certificate. Second prize goes to Harshavardhan Khakre, Mapsu, Nagpur. And third prize is Varun Chekati, SVVU, Tirupati.
Coming to photography competition, first prize goes to Aditya Kumar, Sardar Vallabhbhai Patel University of Agriculture and Technology, Meerut. Second prize goes to Shruti Barbude, Mapsu. Third prize goes to Venkatesh Chavan, Mapsu. Congratulations to all the prize winners. Before we conclude the prize distribution, we would like to inform all the participants that participation certificates are available at the registration counter. You are kindly requested to collect them while leaving the venue. Let us carry forward the spirit of One Health, collaboration, innovation, and responsibility towards human, animal, and environmental well being. Thank you all for being a part of this remarkable conclave. Have a wonderful evening.
I request Honorable Vice Chancellor Mapsu, Dr. Nitin Patil sir, to present the plenary.
So after 6 7 hours of 6 7 hours, yes, 6 7 hours of rigorous sitting and then listening to all these people, I feel that everybody must be feeling very happy and satisfied. And the purpose of holding this particular conclave, especially for the youth and those are uh, bearing the beacon of one health for the veterinarians, that is fulfilled what I feel. I congratulate each one of you who participated in physical mode, those who were present, shown the interest. They are also worthy of appreciation. And I hope that the memories of very good events that were carried out over here and the response from all you people, you must be remembering for the many days to come. And what I hope that in the future also we are going to have such kind of programs, and the people who have been watching these particular events online and keeping us abreast with the kind of alertness that we require for conduct of these kind of programs, so that is also appreciable. And I expect that in future also we'll be having such kind of competitions, and our people, our students, everybody, uh, they are worthy of all appreciations because of their participation. I thank you, the organizers, the participants, and the people who responded to our call on a short notice, and all those people who conducted the quizzes, then the competitions,
and the competitiveness shown by each one of them. So that is all praiseworthy. Thank you again, and I wish you very happy journey for those people who have come over here, and those people who have exerted. I wish them that they should have a very good rest today and tomorrow also. And again, thank you everyone.
About the program. So that can be also telecasted like.
Right.
Yeah, please. Please join.
Okay. Good evening to everybody who's watching. My name is Paransha Adhikari, and I'm from College of Veterinary and Animal Sciences, Rajuvas, Bikaner. We came here yesterday for the National One Health Youth Conclave, and it was honestly a very smooth ride. They had arranged for all our transport also. The accommodation was very good, clean, excellent. The food was also very good, and the way they managed all the students, the form filling, it was very organized. When we came here, we really didn't know much about all the experts who'd be speaking, but I think 3 or 4 experts spoke who are directors of various universities, and it was honestly a very next level experience to listen to them, because they told us about AMR and other One Health issues. And I have never heard anyone speak with so much knowledge about their own subject. So it was very mesmerizing for me. And personally, Dr. Pragya Yadav was very inspiring, the way she spoke, and um, all her history and where she's from. I really liked it. And uh, we actually don't usually get this much exposure, but the exposure this conclave has offered me is definitely going to pave a very important role in what I do in the future, in my postgraduate studies, etc. So I'm very grateful that this opportunity was given to me by the committee who organized this, and they invited us. I'm very grateful. So thank you so much.
Thank you. Thank you. You are from Bikaner or Udaipur?
Sir, Bikaner.
Bikaner.
Thank you, sir.
Convey my regards to your vice chancellor.
Yes, sir.
One more is there. Yeah, one more please. Hyderabad. Sir.
A very warm good evening to everyone. I'm Khushi Sharma from PV Narasimha Rao Telangana Veterinary University. I would really like to thank everyone for making this event such a wonderful success. It was a life-changing opportunity to have come here and represented my college. The quiz was amazing, something we have never seen before at any school level or anywhere. The conduction of the quiz was something that was life changing for me. And we've learned a lot. One Health Conclave has taught us so much about One Health, which we didn't know existed. The scope of One Health was undiscovered for me until today. I would really love to read more on it, learn more on it, and learn more from whatever has been taught. So, yeah, thank you very much for getting us here. Thank you so.
Okay, thank you. Thank you very much.
Thank you so much, sir. I think.